things, some of the analyses you did on Part A, I'm sort of asking for them to be done on Part B again, but it's with a different data set that I simulated, and hopefully that'll be okay. Let's see here.
How long were they in there for? Like a year?
Let's see if there's anything that I want to bring to your attention. Have you watched yesterday's video?
Yes.
Yep.
Okay.
The only thing that I would comment on is it really only $25 for field plot. I know you guys were just discussing it,
Well, John, yeah, right. Jonathan said they were able to do it for less than 20. We're right around 20. And I was wondering if they were really covering all their costs with that. He was convinced they are. What's that?
I said, yeah, exactly. It seems like 25 is pretty low.
Yeah. That's if you, yeah, right. Yeah, that. Like I say, that's about what it is if you've got graduate student, or also known as slave labor.
Yeah.
So let's see. I'm at this point. I don't see. You've had a chance to download these data files, I assume. Is that correct?
Yep.
Yeah.
And the newer ones too, where you added the T1, T2.
Yeah, where it changed.
Yeah.
Yeah, it changed from the. I was. Well, it could have been barley, but barley under pretty low productivity conditions. But anyway, the third trait is probably the thing that, or the third question is probably the thing that you have to think hardest about. And I think Boris said something about, he could even imagine another strategy, and that, if he wants to pursue another strategy and figure out, or if any of you do, figure out what the third strategy might cost and whether or not you'll get better gain for your return on that investment, is certainly, I'll certainly look at that. The thing to, and I'll put a drawing up, not today, because, well, I don't even know if you guys could, I suppose you could see my whiteboard if I really wanted to, but the thing about what happens when you have traits that are negatively correlated is that you can imagine a cloud of data where the slope is going from the upper left to the lower right. So there's a negative correlation. There's a cloud of data around that. And if you're doing your selection correctly, what you're basically doing is moving that cloud of data towards the upper right-hand corner of your XY axis. So even though the cloud of data on an XY axis is following a negative trend from upper left to lower right, if you're doing your selection right, is basically what you're going to do is you're going to move that whole cloud up to the right, and the negative correlation will continue to be what it is. Now, I think the other thing that came out in the class discussion yesterday is I said, certainly trait number 3 as that's non-negotiable. So that doesn't really have to be. In some sense, it's part of your selection index. In another sense, it's not. The index is really on the 2 quantitative traits, and they should each be weighted equally. Is what I decided. I hadn't really thought about it very long yesterday, and I think I'll just leave it at that. You know, that there's each of those two traits need to have equal weight. Now, of course, you'll make more progress on the second trait because you have fewer loci to deal with. You also have the potential to run out of variability for it much quicker. So, but the thinking on this, in terms of you just have to. So you have the data, but you have to imagine, well, what would the data have looked like in the F2? On the F2, all I have is trait number 3 that I can really think about. But, how am I going to select in the F2? In other words, which individuals am I going to eliminate in the F2? And then which individuals am I going to, or individual plants am I going to eliminate in the F3? And I'm going to be doing this with markers, so it's going to cost me, when I start off with my F2 and go to my F3 and so forth. So I'm spending money for markers as I go along. It sort of have to say, and that's kind of now that I'm thinking about it, if you're only spending money on 2 markers out of the 14, your cost per sample is 2/14 of what it would be, at this $10 per line. So there's a couple of little gotchas that I wasn't explicit about in there. You'll have to think through those. Does that make sense?
Yeah, so there's one question I do have is there's never a penalty if you go from pricing out, say, doing 14 markers and you go down to 2 because you reduce it in the number of wells that are on a chip and that they don't ever penalize you?
Oh yeah, they would. And
okay.
Yeah, no, this is an artificial. Well, this is a price per marker, and in some systems, some marker types of systems, that penalties are, when you're dealing with very few markers, that penalty's already applying. So it's the sample size also penalize as you come in with less than 1,000 to make this truly realistic. But right now, let's just assume that the penalty's already there because you're only using 14 markers. Boris, you're a little bit late. I was going to tell you that I looked at your Part A and I started looking at a lot of people's Part A, and yours was it was fun. It was really fun to read. Okay, it was a thing of beauty. So you did a really spectacular job on that. Thanks.
Thank you.
How are you feeling? Are you any better yet?
Yeah, I'm now okay.
Okay.
Yeah. I was since it was raining, I wanted just to leave the university to come home, so that's why I was a bit late.
Oh yeah. Okay. All right. So, is there anything else that we need to visit about for this? Oh, and the other thing, I think I'm caught up with most everyone with grading. There's a couple late ALAs that I'm still kind of waiting on. But yeah, I let those folks know that they still need to bring those in. Is that today's kind of the last day for that. So because I really want to make sure that the grades for Part A are done early this week, so that and all the other ALAs are done this week, so that when Part B comes in on Monday, and you all should notice that it's noon on next Monday, that it's due by, because I really want to get that turned around very quickly because the university has got a very short lead time in terms of turning in grades, because people have to graduate at the end of the semester and all these things. There's all these bureaucratic things that still have to happen. So I want to get it done very quickly.
Are you planning on having lecture tomorrow and Friday at all?
Yeah, I'll show
Okay.
up. Yeah, tomorrow I'm going to talk about. I'm going to essentially review the semester. One of the things. So every year I get kind of a different group of people in terms of their background, and I also change some of the ALAs every year, and so I learned some things from you all, and I want to share with you all what I learned. And next year I will probably reorganize, and based largely on what I learned from you all and in the previous year. There's some things that I can reorganize and anyway, I'll discuss it with everybody, or I'll bring it up tomorrow. Well, maybe I should bring it up with you all now, because most of you won't be there tomorrow during the lunch hour. So one of the things I was thinking about doing is early in this semester, I kind of did some review stuff, at least what I thought was review in terms of analysis of variance and how to run certain, how to generate certain kinds of plots for exploratory data analysis and all of that sort of thing. And I think that what I'm going to do in the future is I'm not even going to bother with that at the very beginning. I think I'll just bring them in, and during the semester we'll learn how to do that as we go. So I want to start off with population genetics right away, and there's spend a little more time on each of those three subsets, and if there are things like learning how to generate cluster diagrams using genotypic data, like we sort of learned how to do here at the end, I'll get that done early in the population genetics section. And again, Boris brought and others brought to me some fun things to do in terms of colors and in terms of presenting the plots in circular ways instead of as things that look like roots dangling from a tree trunk. So anyway, those I'd like to try and just do the population genetics kind of early, and then and along with all of the pedigree information and how do you determine whether or not, or how to if you've got regular sources of inbreeding or if you have irregular sources of inbreeding. Anyway, all that stuff that affects structures of populations. I thought I would start off with that and spend a little more time on it, and not have this sort of dangling thing out front about these various kinds of analyses of variance that you can do. And then go into modeling the quantitative traits and the quantitative trait models and go through most of those kinds of things, but not really deal with multiple environments until and when I do the quantitative genetic models, and introduce simulations at that point, and then start doing some analyses of variance, but not from multiple environments, but from just a single environment. And then after we sort of explore that, deal with the genotype environment interaction and multiple environment trials, and extend the simulations to being able to do that. And then after that, get to selection, heritability, and those sorts of things, genetic gain on a single trait, and then if we have time, get to multiple traits and selection indices. Anyway, that's sort of the way I'm thinking about redoing it, and basically that first set of things that I made a lot of assumptions about people already knowing. I'm not going to assume that anymore, but I'm just going to distribute it throughout the semester. Do I get any reaction to immediate reaction to any of that?
That certainly sounds good. I mean, as the semester shows, we all did pick up on it eventually, but I think that might help ease the next group in. Yeah, the only other thing I can think of, and I don't even know if this really fits the scope of what you want to do, is to see if it could be related to JMP as well. Not because I'm a big user of JMP myself, but just because there are a number of people that do use that program that might be able to get more out of the class by being able to do that. I don't know.
Yeah, last year I did that. I worked with JMP for, about 4 people, I think. Because of the companies they worked for, they had JMP at their disposal. And so I've done that before, and I can certainly do it. It takes me a fair amount of mental gymnastics to swap, switch from one to the other. There's a how do I want to? It's a great suggestion, particularly for exploratory data analysis. My experience was that getting people who are familiar with JMP to explicitly state what their models are is really difficult. They're so used to clicking, and they have the model in their head, and it's all kind of represented as a bunch of clicks on those little boxes that they allow you to put things into the model. But they really never quite got to the point where they could write out their model. So there was this, and I don't know how they do when they move on from a company that supports JMP and start working for somebody that doesn't use JMP.
Yeah, I kind of know what you mean. The nice thing is working with both JMP and R really gives you a good look, kind of what I said, behind the curtain as to what's going on. So you really do understand it. But on a day-to-day basis, JMP is a lot easier
absolutely
to work with.
Yeah, it's really easy, and so you use them both all the time.
Not all the time. I learned to use both of them this semester, but I used JMP because of my creative component.
Okay.
And then R for this class. So.
So JMP was for the creative component, or was there another class using it?
Just for my creative component. It's something that some of my coworkers, and since I'm at the University of Minnesota, it makes me a little bit different, but some of my coworkers have used it, and I was able to use R and get, say, like the least square means and some of the plotting, and then I would do the same thing in JMP and get the exact same thing, so I was confident in my answers and go forward. And if you're a student at Iowa State, you get free access to JMP also. Oh yeah, yep. Okay.
So it's from a cost perspective and this class perspective, it doesn't make much difference.
I just came to appreciate that, just because you have to code in R doesn't make it archaic. It just means that you get to, like I said, see more what's going on behind the curtain.
Right. Yeah.
And understand what's going on.
And most people, if you're working for a large company, you will have access to expensive software. Although, you know, I also, we didn't cover a topic called resource allocation, and what that means is you need, there's software available for that sort of thing where you try to optimize, and we're just touching on it a little bit when we're trying to deal with the trade-offs between cost and time and genetic gain. But it turns out that there are pieces of software that will find the best solution to these trade-offs. Quite often there is a best solution, or a better way to say that there are a group of solutions that are better and for which there are no solutions that anybody can ever find that are better. These kinds of things have been proven mathematically. And so there are pieces of software that you can use, and then you can be guaranteed that among a set of solutions for these trade-offs, that if you choose one of them, you know it's going to, no one can do better than that. They can do other things that are as good as, or design their breeding programs that are as good as yours, but you can guarantee that they'll never be better than yours. And so you know, that whole concept is supported using different kinds of software. One of those other products is called MATLAB, and then there's a bunch of other ones that are like it. And in fact, Excel will do some of this for you, but Excel can take weeks to solve what these other pieces of software can solve in about 3 seconds. Same thing with R. R will also has routines for, and there's a free optimize or piece of software that does optimization, and it's called Goosec, and it's really fun to use. And in fact, we're going to be teaching this topic to plant breeders at Cornell in late May, and we'll have them learn how to use Goosec for that. But to solve some of these problems, Goosec can take, you know, a week or at least a weekend to get it done, and another piece of software like MATLAB can do it immediately. The problem is MATLAB comes with a really hefty price tag.
I would imagine that the hardware requirements are a little hefty as well.
Yeah, and I think even JMP does some of this stuff. And I don't know what the true cost of JMP is, you know, for the university, what the university license is, but you know, it's on the order of tens of thousands of dollars.
Mm-hmm. Yeah, the only other question I have is off topic a bit. Over in Agronomy 523, Dr. Luberstedt's class, he had presented the one lesson on operations management that was, I think, based off of your presentation. And in there, the one question that I kind of got from that overall, a little bit off of the topic of even that, was when you talk about looking for the optimal genotype, and it's broken down into ones and zeros, and you have, say, 10 different loci for disease resistance, and I know that people have broken those loci down into major and minor loci, so a few of them will give you your, you know, a larger percentage of disease resistance than other ones. My question is, has anybody ever looked to see if all 10 loci can be expressed equally at the same time by the cell or the plant, I guess? Meaning, even if the loci are all potentially present, do they physically interact and stop full expression just because the cell only has so much capacity for say, protein production or something along those lines?
Yes. There's it depends on the disease, and or the stress. So something like soybean cyst nematode. There's a gene called RHG1, and it has if you have a certain number of copies, and I forget how many copies right now, but let's just say 5 copies of RHG1, you get a lot of resistance to the soybean cyst nematode. And if you have less than 5 copies, you don't get. But that's not the total resistance package. There are also 4 or 5 other loci that provide resistance, and if you will, it enhance the resistance to some races of cyst nematodes, not to all races. So again, it's also, so it's highly dependent on, in this case, the environment that consists of the races that you're going to encounter in the soil, and it also depends on essentially the genetic background. What are the other enhancers that you have in the genome, and also the number of copies of RHG1. So there's an RHG2 and an RHG3 and 4 and 5, and there's a number of these things. Most people have not studied the question about are there interactions among these, and in some genetic backgrounds, so I have a student that works for Syngenta, and she found that in some genetic backgrounds, there's definite epistasis among these RHG loci. In other genetic backgrounds, they all appear to be additive, with RHG1 providing most of the resistance. So yeah, this seems to be highly variable, and so what's the optimal combination of these things? Well, it kind of depends on what kind of environment you're going to put them in.
Is there any sort of like maximum stacking of RHG1? You said that you have 5 copies. I'm sure people have tried to make more than 5 copies, and to where you get to a point to where you have 10 copies and it's just as good as 5. So you kind of hit your limit.
Yeah, I don't know the answer to that. It's my understanding. I don't know of anybody that's tried to do that.
Okay.
It's my understanding that if you have 5 or more, then you've got resistance. But I don't know, what that really means. Is there a waste of time to do 6, or do you get a little bit better if you have 6?
Right.
This is kind of like Lou Gehrig's disease, and in that case, I think if you've got at least 50 copies of whatever the ALS gene is, I forgot if it's more or less, but I think it's more. If you've got more than 50 copies, you don't express any Lou Gehrig's. If you have fewer than that, then you have ALS, and in that case, it's like a light switch. At 50, it doesn't matter how many less. If you got less, it doesn't matter. You're going to have ALS. So biology is incredible. It doesn't seem to follow rules.
There's a vast amount of diversity in places I don't think people have originally thought there would be.
Right.
Kind of like epistasis.
Yeah. We tend to kind of ignore it. Any other questions? If you have any thoughts or comments on my proposed reorganization of the class, and even suggesting something like, well, do jump and R at the same time, that is certainly, I appreciate it if you have those comments, and you can just send them to me as email if you want.
I guess I kind of have one thing. It's just I don't know what the actual agronomy stats class program used, but I know I took like the generic stats for grad school, and it used JUMP. But I was just wondering if it's worth me, especially for the agronomy one, because it's in the same department, just coordinating and maybe have them use R, because after learning R, I could see it being way easier going to other programs, as opposed to, because coming from JUMP to R was kind of was not fun.
Yeah, that's, we've talked with Ken Moore about that, who's in charge of the agronomy program, the online agronomy program. And he hasn't been very enthusiastic about R, and I don't know what his reticence on it is. It could be just that he's so used to working. So he learned in SAS, and then JMP is just kind of a nice interface to the underlying SAS code, and so it's something that he's really comfortable with, and he's very comfortable with Excel, and for whatever reason, there's just never been any. He just hasn't really wanted to do R. Now the introductory course that the statistics department teaches, is that one that you took?
Yeah, when I signed up for my class, like the agronomy one was full. Yeah.
Okay, so the introductory statistics one actually used JMP.
Yep.
Yeah. Well, we haven't. We did talk to them about content. Because there's a lot of things that we think plant breeders need to know that they don't cover. Things, simple thing, and not simple, but things like cluster analysis and things like at least talking about incomplete blocks, and demonstrating what an augmented design does, and demonstrating what a lattice design does in the introductory class, and they didn't really want to do that, and so that's why we are currently building an introductory statistics class for plant breeders. There's a few other things that we do in there, and it has to do with decision making. So it's really, as well as we introduce people to machine learning, because machine learning is going to become, and prediction, that's going to become such a big part of our lives in the next 10 years. And they sort of wanted to stick with what they had. We didn't really talk explicitly about software. We were just mostly talking about topics that we felt like were going to need to be covered for the future. And they weren't very enthusiastic about covering these other topics. Now, they offer many versions of that general stat class. They have a version for engineers, and the version from engineers has the same number. I don't know what number they call it nowadays. In the old days, it's called 401. But they have all these versions, and so the version for the engineers doesn't look anything at all like the version for agronomists. And then there's a version for educators, for people who are going to become teachers. And that doesn't look like what's for the engineers or for the agronomists. And then there's a version for biochemists and chemists and all. So they already provide a broad range of introductory statistics, and so I can imagine where they were, why it just didn't make, to them it made no sense to do something for plant breeders, because in terms of numbers, there just aren't that many. It's not like, what's that?
Yeah, I took Stat 4-1
Yep.
last fall,
Yes.
and it seems they have changed now because we had the choice between 3 softwares: R, SAS, and JAM. Yeah, so it was up to the student to choose,
Oh.
and whatever you choose, they could guide you. Yeah, so they changed it now.
Oh, so they, at least in terms of what software, now say for your class, did the students have to vote on it, or was each student given what,
No, you choose what you want, and the instruction will
what
help
you
you.
want to use?
Yeah.
Ah, okay,
So we had 3 different software going on in the class. Yeah.
yeah. But the topics were, you weren't being taught topics that were motivated by engineering problems.
No.
Yeah, so you probably didn't get very much introduction to probability theory. Whereas
No, not so much.
the engineers, they get an enormous amount of probability theory. So yeah, so we will end up teaching something that is built around the concept of decision making, and that is much more like an engineering kind of class, but it won't include all of the partial differential equations and those kinds of things that engineers are familiar with.
That will be good. Yeah.
And since you said that they let every student pick which software they want to use, I can see doing that for my class. It means like I say, last year I had to learn JMP, and it's it's okay to be bilingual.
Yeah, true.
So they had JMP and R, and what was the other one?
And SAS.
And
SAS
SAS.
is
Oh, okay, all right.
yeah, that was Philip Dixon. Yeah.
Yeah, Phil Dixon. Yeah, he's very good.
Yes.
But yeah, he's also worked with biologists for a long time. So
So he's already bilingual, that's what you're saying.
yep, trilingual. Yeah.
Yeah.
Well, I was thinking where he can speak math and biology.
Well, I don't know. Phil is not a theory guy. He's an applied guy. So, and he's worked with biologists and mainly with ecologists throughout his career. And so, spatial statistics and autoregressive kinds of models are things that he's really good at. I think he's originally got his degree originally from the University of Minnesota.
Well, the U of M's good at the engineering side. I know that.
Yeah, Phil was not coming from that direction though. He was
Okay.
definitely coming from, forestry and ecology.
Oh, yeah, forestry. They're big on modeling there. I know you said that he's more of an applied guy, but
Yeah, he's definitely an applied guy, and trying to the big deal is try to sort out when you transition from one habitat to another. These things, and so these spatial statistics are things that he's really good at, which are really good for plant breeders in some cases, say iron deficiency chlorosis in the Red River Valley of Minnesota and and North Dakota. There are
that's my old stomping grounds.
yeah, so there's a bunch of you can call them habitats where you have these big iron lenses that are running through the soil, and they create all kinds of, or actually the iron is all chelated, and so it's not available to the plant, and it creates all kinds of problems. And how do you tell when you're in one iron poor habitat and when you're in an iron healthy habitat?
Yep, and that's a big thing up here in the Northeast where we have the iron mines and
Sure.
then and wild rice in its natural habitat, and how iron in what form it is and how that affects sulfate to sulfide reactions, and
Yeah,
that can either be benign or it can be toxic.
yeah,
It's interesting.
yeah. You don't want to drop a well in the wrong place.
No.
Yeah. So anyway, yeah, that's the kind of thing that he's very good at. That he's worked with biologists for a long time, because human eyes are not really good at discerning what the plant and the animal species can tell when they're in a desirable habitat and when they're not. Anyway, that we wandered a long way off. But that's I think I'll take that to heart, and maybe next year say, well, if you want to use JMP or SAS, that you pick. It's okay with me. Any other reactions? Immediate reactions?
I like that it sounds like the learning curve will be smoothed out a little bit, because this class in itself can be intimidating, let alone having to learn all the coding at first. So I think it will be a little less daunting off the get-go in the future.
Okay. Yeah. If we can. Yeah, I agree that having to learn the concepts and the theory at the same time as you're trying to learn coding that's a bit of especially if you're working full time.
Yeah, well, it just took away from it sometimes, where like I didn't spend as much time really thinking about the concepts, because I was just like trying to make the program do the tricks I needed it to do. Yeah,
that's a good point. Yeah.
And that's something, I'm thinking back to what Boris was talking about yesterday using PopVar.
Yep.
We used that in 523. I was just helping out a couple of other students to use that program yesterday. And, because they don't use R and weren't really familiar with it. And I believe they said that they used Jump in your class last year, so you probably know who they are. But anyways, so and one of the things is PopVar does take some time depending on how big your data set is. So there was a little impatience, I guess, with it.
It's busy simulating huge numbers of progeny to figure out who's going to have the best, which population or which crosses are likely to produce the best or most desirable families. That's that was originally PopVar was originally written by some guys at University of Minnesota.
Mm-hmm. Well, I thought that it was really good in the fact that maybe some of those programs do need to be around, because as a breeder, we need to be a little patient sometimes.
Well, there's kind of part of what we've learned with, the millions of man hours that exist in something like Excel, is that we have a high threshold for new software, and so you have some guy in his garage that, or on weekends, develops PopVar, maybe 2 or 3 days, and they maybe put 1,000 hours into it, and yet we have this expectation that comes from things like JMP that have been around for and have got literally millions of man hours invested in them. That's for people who are just doing this on the side. That's a very daunting situation to be in.
Mm-hmm. Well, and that's the one thing that I've kind of noticed getting deeper into the plant breeding world, is that there tends to be kind of people who are more patient, and then there are those that want their answers right now, and we should be able to get them. And sometimes you got to wait for the plant.
That part too. And I don't want to wait for the plant and a piece of computer software.
Yeah.
No, that's just part of the reality. Anything else? Okay, well, I'll see a few of you tomorrow at noon, and then and again Thursday, and hopefully by tomorrow a few of you will have made an attempt at Part B, and we can iron out any questions that you may encounter at that point. See you later.
Yeah, have a good night.
Okay, bye.
So,
Yes, bye.
Who's ready to get started?
I'm ready to do this. I'm also ready to eat.
Well, as long as you've got your priorities straight.
I guess I could do this for a bit before I eat.
Well, no pressure, jeez.
Yeah. Lots of calculations, I guess, if we just like start going through number one. I only like looked at the assignment. I haven't done anything. I changed the font because I don't like Times New Roman.
that's the important thing.
I was going to say, as long as you did the important stuff. I'm just basically been working on making out the table in an Excel spreadsheet.
I looked at the questions. I haven't messed around with the data set at all yet. Or data sets, I should say. I guess there's many of them.
Yeah,
So-
I haven't either.
In the chart, isn't the light blue supposed to be the average number of cysts and the dark blue numbers, like the number of individuals with that genotype?
That's what I was trying to figure out too. I was trying to figure out if it was that or if that was the average number of cysts for a plant that has both of those genotypes at both of the loci.
That's what I was trying to figure out yesterday, and then
And you didn't ask the question? Oh,
I just opened it.
I don't have the answer.
Well, why not?
Well, doggone it, I was chatting about unimportant stuff.
yeah, I got very distracted while he was on here.
Well, just looking at the way the numbers work out.
Yeah, because it didn't seem to match up with like the frequencies.
Yeah, that has to be average count of plants with both, right? Or you just said it doesn't match up.
Well, I thought that I had tried it yesterday and to figure it out, and like, yeah, that it wasn't matching up with the frequencies. If the
Well-
was the number of individuals.
Oh yeah.
Well, it says that like genotype, frequencies, and phenotypes are summarized in the table. So I wonder if that's like it says average, but if that word might just be confusing.
Didn't he also say something in lecture? I was going to go back and try to listen about having more phenotypes than genotypes or something like that.
Yeah, and that's in his files. Like there's, I don't know, I think he cut down the phenotype one to like 4 families, but the others have like 6.
Okay, I couldn't remember if that was like some of the other data he had or if it was referring to this.
Okay, yeah, and then it makes sense logically too, because you can have similar you can have many phenotypes with related genotypes. But that's not important necessarily.
It just seems like too, if the dark blue is representing how many cysts there are if you have both genotypes, like 31 seems low if like just big B by itself is 107 and big A by itself is 150.
Yeah.
Yeah,
those
that'd be
numbers don't
disappointing.
make sense together at all.
Well, if they're additive, though, I mean, that could make sense. Because, hang on,
If you have big A, little b, you have. But then the numbers get incrementally bigger as you move. Like 377 and 383. Oh, not 383. 377 in the one corner, though.
I guess big A and big B are both supposed to be more resistant, right? So I guess maybe it does make sense that it'd be quite a bit smaller when you have both of them.
yeah,
together.
they're.
Yeah, basically what I'm seeing, tell me if I'm wrong, is that the B locus genotype, the small B is more common. So call it susceptibility, but heterozygotes and large A are more common on the other end. And so I think that's, that has a tendency for non-susceptibility. In other words, they're negatively correlated. Maybe. That's just a pattern I'm seeing there. There's a tendency, if we ran a histogram on it, there's a tendency for the large A to be larger or more represented, and the small b to be more represented. That doesn't make much sense. I'm having an off day.
So
No, I know what you're saying.
it seems like
Yeah.
big A with little b, it looks like it'd be like the most common frequency. Or...
The most common should be big A, little a, small b, just by the frequencies.
Yeah, that's what I'm saying.
But I suppose what well, they're saying the average number of cysts. But what you're seeing is that big, small a, small b has, I don't know.
So,
It's really susceptible. Well, that's all I'm saying. Huh.
I guess. Yeah, my brain is slowly turning off.
Yeah, it's been a day ever since the fertilizer showed up a day early and at noon. So it's scramble. This day just kind of went off the rails there and never recovered.
Cool. Okay, so, I guess for part A, does that mean average cysts per individual in the population? It's gotta be what it means, right?
I don't see anything else you could base it on.
So it's just 100 times the average times the frequencies for the two loci that go into that average, right? And then you just do that for each of the 9 boxes. Is that no? Yeah, because that would give you total cysts for all the individuals with that genotype, and then you would add all those up and divide by 100. Feel free to tell me if that sounds completely wrong. So Kyle, you freaking nerd. That's wrong.
Well, I say let's give it a shot, Kyle.
Man,
See what happens.
That's like too positive. I don't know if I like that.
Hey. This, regardless, this is it, guys. This is the last thing we have to do, and we're done.
The final assignment. Do 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 do.
Well, it's an exam, but it feels like an assignment, but it's an exam. And it sounds like he's pretty positive about the first half, so we're kind of already like half golden.
We got at least 50%.
Yeah, so.
I would be lying if I didn't say that I calculated out my grade. Not just like exactly how bad I could do, and I did not have to do very well.
Yeah, I did the same yesterday.
I haven't really calculated it out, but it was good enough that I was like, I could not write it and not wow.
If I would have got a 0 on my assignment, ALA 15, because that grade wasn't in yet yesterday, if I would have gotten an 80 on the final, I would have gotten an A in the class. If I got a 75 on the assignment, I'd get a 62 on the exam. I'm like, very difficult.
On ALA 15?
Yeah, I turned a 15. I turned it in like a day late, and then it got lost.
It got lost, he says.
No, it got lost in his email. Like he legitimately came back, and I was like, "Oh yeah, I didn't see that."
His computer ate your homework.
Yeah, dude. It's not fair. I did everything right.
Yeah. Anyway, so you were talking about how to calculate the population average. Is that what you were just saying before?
Yeah, I think so.
That sounded very complex.
No, it's here. Give me a couple more seconds, and then I can share my screen and I can show you like with the functions and everything.
Okay.
Only a couple seconds, Kyle.
It'll probably be more than a couple seconds. You know.
This table is linked to this text.
Oh yeah, you have to like basically just create your own table.
That's why I was doing it.
Now I understand. I was like, what Excel spreadsheet work are you doing?
I'm sure it could be dealt with in other ways, but I'd rather just make a new table and be done.
Yeah, that table's some weird format. If you click on the blue numbers, it says they're part of like a function. So I was not about that one-ish.
Table properties.
So what you're saying is the programming or statistical life didn't choose, or you didn't choose the statistical life. The statistical life chose you.
Yeah, pretty much.
Something like that.
All right, let me share my screen. First, I gotta pick the right screen to share. There we go.
Yeah, that's always fun.
Alright, yeah, see this?
Yep.
I didn't hear what you said you were going to do, but I did what I thought might work, and I think I got the exact same number as you.
Yes, so I was just taking the frequency times frequency of both, so times the count or average yeah, average count for those loci combined times 100, because that gives us total. It gives us total cysts counted for that combination of loci. If that makes sense.
Oh, you already split yours up different than I did. Yeah, but I'm dumb, so don't.
Well, I just went like the 0.09 times 0.36 times the 31, and I did that for each of them, and then took the sum of all of those, and that equaled like the exact same thing, I think.
Yeah, you know, I just realized that doing it times 100 was completely pointless because it's already an average.
Yeah, so I have the exact same number as you, doing it that way.
148.
Yeah, 148.4156.
All right, that's cool.
See, I did the thing to where I put the BB and then the 0.09 in the parentheses with it instead of making it available to me.
Oh.
Feeling kind of stupid.
I started to do that, and I was like, wait a second here.
I was thinking that too, but I'm like, no, it'll be okay. No.
Just plop that right in there. Boom. 1A, done.
Well, look at you go.
Swag. Average for the A loci.
All right.
Well, that would be
So-
similar. So,
Man, I've got to learn my Excel foo as good as you guys do because
pretty much if
organizing is not my skill.
it's not field season, I'm working in Excel almost constantly.
Yeah, my colleague, my buddy, who is in forestry and forest modeling, so I understood what Dr. Beavis was talking about there, is always working on this stuff, and he's the guy I go to. I'm like, how do I do this? I know Excel will do this, but how do I do this? Oh yeah, it's real simple. And then he shows me, except I work on a Mac. And he's like, I hate Macs.
Sounds like a personal problem.
Well, he's not wrong. It does things that you don't, well, there's certain functions actually in Excel that it won't do.
Yeah.
It will not do them, and he's just flabbergasted. But, so I've kind of been lazy and I can get away with not organizing it.
You know, I would really like if we didn't have to, words. Didn't have to make these all separate answers.
Alright, let's take a look at what you're doing. Because you look smart.
Fooled you.
Not really. I noticed everybody's on here. Everybody must be excited to get done with this.
I actually left work a little early. I was like, I was like, I gotta go. Like, I gotta be done with this class.
We'll be better employees when we can be more focused.
I mean, yeah, sure.
Yeah. Not saying that there's anything wrong with the type of employee that you are now.
Let me know if that's what you guys are getting for your average.
Are you still taking it times 100 for your answer?
No, because that was pointless. Because basically, like, I had times 100 on all these individually, and then you just divide by 100 at the end anyway. Because I think who was saying that they didn't do 100? Sorry, I'm terrible with voices.
I was saying that, and I got the same numbers for you as you for A and B as well.
Yeah.
Added header? What?
like this so far. Is this the whole thing, I hope?
What, this set of easy answers?
Yes.
Maybe. I mean, it does. I looked through it, and I wasn't like, I'm totally stumped and I'm gonna die. I don't know what I'm doing.
He kind of said, like, question 3 is the tough stuff. So I could look.
Well, at least we're getting the same answers. I'm happy about that.
Whoa. It's weird. Whoa.
What are you
This
doing?
table is like moving around on me.
Yeah.
I have noticed that.
One.
I have noticed that Excel has been doing weird things with this class, so I'm wondering if there isn't some sort of Chinese bug in here.
No, it's not Excel. It's it's in the Word document. What the? It's like tied to this section. Get out of here.
My.
No, yeah, I noticed that problem yesterday because I was playing with it, and that table just keeps shifting things around.
Yeah, like, I don't know what sort of weirdo formatting stuff is going on, but I don't like it.
You know what? I'm gonna pull rank. Get out of here, table. My table's way better.
Oh, is that the table you brought over?
No, that's the table he had in there.
No, that's what I mean. Did you import that into Excel or did you create it?
Oh no, I just hand typed
One
this in.
Okay, so that's not causing a problem. Oh, you imported it into your Excel file. Got it.
Yep.
Got it.
Well, I'm actually copying this and putting this table
Yeah.
into the Word document because it's the one that's in the Word document is super wonky.
Yeah, that's what I was gathering, but at first it sounded like you were going to do it the other way. And all I can see is your Excel file, not your Word document.
Sorry. I'm living on that.
No, that's that fun thing about Excel or Zoom.
Living that
That's fun in the past.
living that dual screen life, man.
Yeah. If you aren't having fun with the not being able to share the files properly, then you're forgetting to turn your mic on and or be close enough to it.
Oh,
I
yeah.
keep my mic on off all the time. And then I'm like, wow, they really don't want to answer my question. It must be so stupid. And I'm always terrified of, I'm glad I don't have a headset. At work, I've had that, and I'm always terrified of forgetting it on, and then,
They can hear you making comments.
Hey, question, Kyle, how did you do your individual ones? Mine came out slightly different. Okay.
Yes.
Yeah.
I just did frequency times average.
Okay.
Is that what you did?
No, I was looking at the original table. That's what it's based off, not just the.
Oh, you're looking at this guy?
Yeah.
Okay. Yeah, let me know if
That's
this is not what somebody.
Well, it makes sense so far.
Yeah, no, I think what you did made sense.
Okay.
Yeah, you're not wrong. How's that sound?
Hey, you know what? We take those. It's all about the small victories.
So the question about your part A. So did you rounded yours or multiplied yours by 100, and is that why yours is like 100 and 319?
Yeah, so here, I could technically.
Yeah, get rid of that. Get rid of it. Make an executive decision.
Okay, good. I was doubting myself because you'd mentioned that was the thing, and then I looked again and I was like, oh God, do I need to change it?
No, it should end up with the same number. Here,
Okay, and yeah, they were the same numbers. It was just a rounding thing. I was like, but why am I wrong?
yeah,
But I guess it was
same number.
just my own self-doubt was the problem.
I just made it way harder on myself by typing an extra 4 keys per formula.
We each have to have our problems. Yours is taking it times 1,000, 100 when it doesn't need to be. All right, so Your average per plant. Woohoo! Comparable to your overall count.
Wait, what is that's for D?
No, I'm just looking at your overall count and average per plant, and they're the same thing. That's all.
Oh, it's because here what I can do is this. Look at this. You ready?
Yeah. How come mine is different than yours?
How
All my
different?
numbers are the same.
This is different.
Okay. Yeah. Go there. Sum of. Let me double check here, but I've got the exact same code as you, and I've got the exact same numbers showing up in my box, but my sum is different. Mine's 144. That's bizarre.
So these are all the same.
Exactly the same, and they're to the exact same length of numbers.
Weird.
Hang on. I thought that might be it.
Okay. Now yours is lower than mine. What the?
That's because I took out that top row. What was yours again?
Mine's 144.0308. Now, I haven't double checked all of mine, but still, I like what you're doing there.
Yeah, it's getting super close, but it's not.
Okay, can you do a,
I mean, if you just
Quick
average like the original table, it's 144. But I have the 148 as Kyle. Point
Yeah.
everything.
okay, so let's, what the heck? I'm doing the same calculations as you to get the same exact numbers in that setup. Yep, C4, well, mine's,
It's probably a different order, but
yeah, but it's still, yeah, you took your B times your A, I took my A times my B. Woo. Anyway, but they're the same numbers. Getting the same numbers in the boxes. Summed it, now I summed up each row, BB row, and that, I did a sum of each of those and ran it down. Let's see what yours is. Oh, your middle one is different. Why in the heck? 23,582, 13,2. Oh, well, that might be it right there. Your BB. Your big B, little b, AA. Yeah, that one right there.
Okay.
The 9. Oh, my box got moved. I bet you that's what it is.
got him. I saved it.
I bet this will fix it right here. Yep, that fixed it right there. My mistake. So at some point in time, I must have moved it. I was doing it on the 31 instead of the 60. That was it. Let that be a lesson to you kids. Make sure you know where your boxes go.
So
Alright.
for question B,
Wait, what?
or for question 1B,
Oh, okay, yeah.
how are you calculating your AA?
So I just did because that's the average for that loci overall. times
Oh,
the-
so it's just those two numbers together.
Yeah. I got it because that's basically doing the same thing as the other one, just not taking B into account at all.
Yeah, I see it now. I was making it more complicated than it needs to be, which is the name of the stats game.
Pretty much.
Well, where's the fun in making it simple? Come on.
That doesn't impress people at dinner parties.
Well, yeah, you make it too easy, and all of a sudden everybody can do it. And who cares about that?
Okay, and then you can just average across the whole thing. I should just like check.
Some of them.
Oh, some of them.
Yeah. And when I first did this, I thought, well, those don't average out to that, but it's because they're weighted together because it's different frequencies.
Yeah.
What the? And this, every time I do stats, my computer starts doing weird stuff.
Like stats.
No, yeah, no kidding, huh? No, I've got a media card from my camera that's just sitting in there. It's been plugged into my USB port for the past day and a half, and all of a sudden it's like, oh hey, you've got this. You should look at it. What? Okay, so we're down to D.
Yeah, this is where I'm not sure where to go for this.
A little trickier.
I think that's supposed to say non-epistatic genotypic values, because otherwise it's kind of weird.
What does it say? It does say it's
It says-
non-epistatic.
It says on.
Oh, maybe I changed it to non already.
I just downloaded mine. It says non.
Okay. Well, mine now says non too, but it said on epistatic genotypic values a minute ago. Ooh.
Rather than off epistatic?
Well, I'm not sure.
That was a bad joke. That's all.
It's okay. I'm surrounded. You would like my eldest daughter, Cassandra. She not only enjoys drag and cosplay, but she is a horrible punist as well.
She sounds like one of the brightest minds of the future. I feel good about it.
Puns are the height of comedy, okay?
Yeah, but for those of us who have to listen to it constantly, it does tend to get old.
I mean, Will Shakespeare wrote puns in his plays, so you know they're good.
Yeah, but Shakespeare was a plagiarist, so.
Hey, you know what?
All right, so actually, the first
Okay.
one's asking for the population average. Is that the same thing as the average per plant?
Hey, yeah, right? Because otherwise, what would your population average be?
No, I'm just.
You don't like my label?
No. Just one of those things I had to ask last in part A about whether or not the entry mean was the same thing as the population mean or something like that he was asking for?
I'm posting
Or entry
this.
mean level.
I'm putting this into my exam like that.
Oh, yeah. Okay, you stop.
I guess that's more accurate.
Are we giddy tonight to be done or what?
Yeah. Very unfocused. Week of rain has basically completely ruined my work plans all week, so.
Oh, no kidding.
Where are you, Kyle?
I am just west of Iowa City, where we're.
Oh, okay.
Yeah.
So you guys are getting rain while we're getting snow up here. Yay!
Ah,
So
you can
Cassandra,
get that.
thanks for that. Yeah, thanks, Cassandra, for sending that down.
I didn't get any of it. Talk to Chelsea.
Yeah, I know. That's kind of what I mean.
Oh, I don't control the way that westerly wind patterns work.
I don't believe you. You're Canadian. You guys are in charge of everything, aren't you?
Well, that's racist, man.
Yeah, more than racist, let me tell you. And I know all their secrets because I'm their neighbor.
Well, we're pretty friendly neighbors, but maybe we'll stop inviting
That's
you to the dinner party.
I've never been invited, so man. Anyway.
Non-epistatic genotypic value.
So for B and C, I am wondering if it should be the average and not the sum, because he's asking for the average. And I'm looking at the quantitative genetic models notes, page 18, it's kind of showing like it divided by 3. And I'm trying to figure out, because it says calculation of single locus genotypic values is what it's averaging.
Yes. That would make sense. Like, I'm not sure why you would sum it across, actually.
Because those are already averages.
Yeah.
You want to take an average of the average? Wow. Are we
Yeah,
gonna be
because-
dividing by 0 and opening black holes too, or what?
Because if I would have times all of these by 100 again, like here, hang on, let me just do this. This will be our little test, and it's broke. And he killed it. All right. So here we'll do this times 100 plants, right?
Yeah.
And that won't drag over nicely.
Of course it won't.
So that gives us that, right?
Verifying number. But decimals. Yeah, okay.
So it gives the same number at the end. It's
Yeah.
it's like what, over here, like I did this with times 100, and I think it was Chelsea,
That's Cassandra.
Sandra, dang it, sorry, did it without multiplying by 100, we ended up the exact same thing, just with one less
Oh,
step.
no, sorry, that was Chelsea. You're right.
Okay, if you want to stop sharing, I'll share the notes. And even if this isn't, even if we don't change anything, it's got the epistasis information. Okay, can you guys see it?
Yep.
Yeah.
Okay, so, because right here he's saying like, well, and I'm thinking single locus genotypic value is what we're finding for A or B, and he's doing an unweighted average across the genotypes here. So it's still an average if this is exactly what we're calculating.
Yeah, you've already done your averages, now you're just summing up your averages. Right? Is that the layman's way of reading into this?
So,
Yeah, I'm just trying to figure out how this table worked, if this was really the average, or if he was still averaging what he had here. Because right here, it looks like he's just averaging this. I'm just trying to figure out how all this came together.
so those numbers that are in there are like what the individual counts for that loci? Is that what that?
Yeah.
Okay.
So I guess then we did frequencies off of this instead.
I think that's kind of like because he's just each one is weighted the same, whereas we weighted them by the frequencies that we were provided.
Mm-hmm.
So I think that's like he divides it by 3 because they're each weighted the same, but we multiplied them by like the 0 point whatever. I don't know what they were.
Yeah, I'm just wondering, because we did weight it, but do we need to weight it, or should we be doing it off the unweighted?
I don't think so because we have those because he gave us the frequencies. So that would be like that would tell us how much of the population had those that average counts towards.
Okay. The other part, so
Alright, is that what he's asking for? Is just the counts for the A locus, right? I don't know what else you'd be asking for at that point. Because he doesn't mention genotypic value
until later.
yeah.
Yeah, no, that's fine. I was just going through the notes and
No, that's a good.
trying to.
No, I sound super St. Louis saying this. Oh, that's a good call out.
But yeah, so here was where he's calculating the non-epistasis and the epistasis values. So I still haven't quite figured them out, but
That's in the notes.
yeah. So I can stop sharing if you guys want, or if anybody's looking at it.
Does it say more about like what that
No, so like
or what those represent? The G, IJ plus G, KL minus.
I can go down.
I'll open it up.
Yeah, no, there's a little bit right below it. So using these calculations, population average. So yeah, I mean, I think this is actually right, because this is what we just did, genotypic value by frequency.
That's on page like 11, right?
19.
19.
I was messing with you.
So no, yeah, I would check that, and then we can.
So do we all have, okay, let's do this. Anybody want to take bets on when we get to something that is totally screwed up? And we're all like, can't figure this out, or there's 10 different ways to do it or something?
There
Second
has
of all,
been a streak of that.
yeah, I'm only bringing it up because, okay, for A, do we all have the same population average of 148.4156?
Yes.
Okay, for the average for the A locus, do we have 193.52?
Yeah.
Yeah.
And the B is 265.83?
Mm-hmm.
Yay! We're all good so far. I just thought it was hilarious. I'm sorry, when he was talking in class about, and then also with us on Sunday, about how, yeah, some of these things act kind of weird and how they interact. And so I'm like, oh, you didn't go through all the genotypes, I guess, or the markers. So I loved that one that had a 100% negative correlation. I'm like, yeah, that could probably screw with our answers a little bit. Anyhow, sorry.
Okay. So for B and C, we're decided.
Yeah, I think we're good there.
I think
We'll
so.
leave them as sums rather than average.
Michelle, I see what you're talking about, because further up in the notes, like he does have unweighted marginal means and things like that.
Mm-hmm. And no, I mean, I haven't looked at these notes in forever, so I'm just trying to interpret.
If it makes you feel any better, I haven't really looked at these notes ever.
Ooh, an honest man. I don't know if we can handle that tonight.
Yes, we can.
I'm willing to say I've looked at all the notes and mostly forgotten them. So,
They've given me trust issues.
now, let's not go there just yet. We know what you mean, but
Non-epistatic genotypic values. How do they make you feel? What are you thinking?
yeah, I have trust issues. It's not a good
Because now, I'm looking at this. I don't think. Yeah, I don't think what I did is right. I think Michelle's right.
I'm trying to work on figuring out the example. Do the same thing and
Yeah, because on page 16 there, Michelle, that bottom table, bottom right, that would be your population average. Right?
yeah.
Yeah. So it's just divided by 3.
He
That's
doesn't
just
use
too easy.
he doesn't use averages in any of those charts, does he? They're all based on genotypic values, not averages.
Are they based on genotypic values?
That's I Just like skimmed through it, but I thought they all
Oh,
were, like,
yeah,
this
it is.
table 4.7.
Yeah.
There's always a gotcha, I'm telling you.
That would make sense, though, if that's how we're supposed to do it, because that flows right into doing non-epistatic and epistatic.
What, the way you did it, or the way you're considering doing?
The way it actually looks like you're supposed to do it. Using by context clues.
Oh my.
Hey man, we're all nerds here. That's okay.
I was thinking more like you're starting to lose it.
I think I lost
like-
it once I signed up to get a master's.
I was gonna say, you're about 3 weeks too late compared to the rest of us. I lost it with ALA 15, I think.
No, ALA 15 I gave up on Sunday night and I just turned it in on Monday.
And you still did good.
I did okay.
Well, nobody did great from what I understand, but
I think I got a 17, so I'll take that.
better than a lot of us.
Average for the B on Lucas. So
So nobody took any bets, but apparently question 1, subsection A is going to, or B is going to be our place. Got it.
yeah, okay. Yeah, I'm like 100% on it. I'm gonna change mine.
To averages instead of sums.
Yeah, just like averaging across the three.
For both A and B, right?
Yeah, I think so. Because it's I don't know.
Well, here's my question. If you're using the average number of cysts, then that's already an average, but I think that those 9 squares, those 9 in the middle are the raw data, if you will. And so maybe it just depends on what you
Those
use.
would still have to be averages though, right? Because the raw data would be, you'd have to have 100 numbers.
Yeah, that's an average of all plants with that genotype. And yeah, I know in the notes it says it's supposed to be a genotypic value.
Yeah, because I mean, like, he does have the population average value can be calculated by genotypic value times genotypic frequency.
Population average, you said?
Yeah.
That's in the notes here.
Yeah. I'm looking at it on page 12. Just got it in a couple different places.
Oh, I see. Hey, yeah, that's what I did.
But we don't have the genotypic value.
Yeah. Maybe that's what we're. Is there a section here earlier on about calculating genotypic value? Like just going off this? Not good. There's only 47 instances of genotypic value in this paper. Here. I think. Like, I'm not crazy, right? Like, we haven't done this before.
Oh,
Well, there's a difference between us having done it before and us having done it before correctly.
yeah. I mean, I'm almost thinking like for the population average, or like the allele average for sure, we leave it as is, I think.
For the allele ones? Just
Yeah.
like average. Because I scrolled to the bottom of those notes you had open, and it just straight up has average at locus A, average at locus B. It's two of the last things on the thing of notes.
Okay.
Just population. Oh, good. There's only 41 instances of that. Population mean. I've been in stats before. Mean can also mean average.
Yep.
Well, that's single loci though. Alright, so the only thing I've seen so far to go off of, in the class notes under single loci for population averages, it states, let me find the exact thing. I know we're working with 2 loci, but this is the only thing I could find. To draw inferences about genetic properties of a trait, we model phenotypic values using linear components. The most common model consists blah, 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 blah. This is usually written as P, or phenotypic value equals genotypic value plus non-genetic component. If we assume that the sum of E is 0, then P equals G. So I think him giving us the average cysts per loci, or per genotype, is, we're supposed to use that as our G. You know what I'm saying? Because technically, let's see.
Yep.
If that makes sense, I don't know. So yeah. So that means for this one, our heterozygote value is plus D because it, well, wait, no. Gotta think of that in inverse. It's in between. Never mind. But it deviates towards the homozygous dominant, so it's just pop D, not plus D. Make sense?
Whoa.
That was scary.
Sorry about that. There's Excel yelling.
The only thing I'm not sure of is since since recessive A or recessive B, whichever one, is allowing more cysts to form, does that mean its genotypic value should be a negative, or do we just go with it at face value? You know what I'm saying?
I'm saying whenever we're left alone, we come up with more questions than ever when he's around.
I mean, yeah,
That's all I'm saying.
we just need him to like sit in the background, log in as somebody else, and just listen.
Yeah, yeah. I think he may. That's the scary part.
Hey, that would honestly be
Is he Lawrence? No.
no. I actually.
Well, I thought it was kind of interesting he popped in on Sunday, just kind of unannounced.
Oh, that's funny. No, I know Lawrence is real. I saw him ask some questions about R in one of our work
Oh, yeah, yeah.
channels.
I believe Lawrence is real.
Or did I? Or is Lawrence a deep plant?
Yeah, yeah. Because this class is so much of that we have to do it that way. That would be fun, though.
That would be more pure than any survey.
Well, not just that, but it's also just kind of, I've been teaching, Dr. Beavis, I've been teaching for 30 years. I got to spice it up a little bit. I'm going to pretend to be somebody else. Just to throw a little spice in the mix.
Okay. I'm almost formulating a thought.
Well, so far I don't have anything extra to add to the mix, but I am digging through old assignments.
Okay. Okay. So this is taking me longer just to type out from all the subscripts and all this crap than anything else. Okay.
I gotta step away for a minute.
Yeah, so the more I'm looking at this, 100% what I had earlier, all garbage. Don't have that as your answers.
What we did earlier is trash.
Yeah,
It was a warm up.
it was a warm up. Exactly. Actually, oh man, what if this ends up just equaling the same thing? You're like such a ding dong. Yeah, whatever. You figured out this. This plus. Well, that didn't work. Why didn't it work? Valid cell reference error. What do you mean? Oh. Okay, I think I did it. Has anyone else made any headway?
I'm working on D and E. I think I have them.
Yeah, I was kind of going through the whole example and think I figured out how to do D and E also, but I didn't redo the others yet.
Yeah. So I've only got A redone. It took a little bit. You gotta.
So what are you going off of now?
Like where in the notes I'm going off of?
Yeah.
Page 11.
11.
And this is for just the whole, like going back to A.
Yeah, this is like starting back over on A.
Okay, perfect. Page 11.
So page 11 and 12 is what I used. So we're using our genotypic values that we have here because we're like assuming, that there's no environment, or that the the sum of environmental changes is 0, or impact is 0. So yeah, so to get your mid homozygote genotypic value, it's you average any of the combined loci genotypic values that are homozygous for both loci, and then average that, and that's your MH. And then you have to calculate your expected values of GA and GB, which is on the bottom of page 11. And to do that, you have to calculate all the other stuff, which, if you go to 12, right underneath the table, that actually gives you like a little example of how to calculate all those different variables. So
I was
basically,
afraid of.
yeah. So I just calculated it, and yeah, and typed that whole thing into Excel in one function, and it worked. It was super fun.
Alright. I'll stop doing the easy part.
So
The
it is.
epistasis and non-epistasis really isn't bad.
Oh, really? Compared to that? Yeah.
But that seems pretty easy to figure out.
Population mean equals Y equals MH plus vector values of GA and GB. Oh, okay. I'm looking at that.
But it also says the population average value can also be calculated as genotypic value times genotypic frequency. And the sum of genotypic value times genotypic frequency. Isn't that what we did?
That's
If
what we did.
genotypic value is equal to phenotypic value?
Yeah, but we got a different number.
But I'm just saying it says that on page 12.
Yeah, I saw that too. I don't know. Where's that? Where's my page? Here we go. Genotypic value times genotypic frequency. And that's what I did though, right?
And then it's the sum of those, which is what we did originally. And
Yeah,
they're saying
but,
the phenotypic value is equal to the phenotype.
so I'm not sure if something happened wrong there, but for doing it this other way, I got 127. The other one we had 148.
Yeah.
I just feel like the way we originally did it makes a lot of sense.
So you guys figured it all out, right?
We're maybe less lost, question mark.
We have paths that we're at the fork in the road, if you will. I'll be right back. I have to grab my soup.
Because Kyle is saying here on like 12, they use the equation that it's provided, but then it also says the population average value can also be calculated as the sum of genotypic value times genotypic frequency, which I thought that was what we did originally, but it's two different numbers that you get, so I don't know.
Well, what do we want to try and argue? Well, what are we trying? What do we?
I mean, I saw that too when I was working through it, and I was hoping that it would end up being the same number. But it is different. So Jacques, for calculating it out, I got 127.35.
If you do average instead of sum.
No, like going through
No,
the
you're doing the different
calculation.
Doing the whole
Yeah.
different calculation. Okay.
What did you? How did you get the D value?
So the D values are your. Let me see here.
Can you share your Excel sheet? Is that where I'm assuming that's where you did it?
Yeah.
Might be a little easier to.
Oh, this one. Here we go. All right. Can you see this?
Yes.
All right. So your D values are your heterozygote
Yeah.
minus the average of both your homozygotes. That's what your D value is. So like D of A is only the A's, obviously, and then D of B is those guys.
Well, that certainly makes more sense as far as that's what we've been kind of dealing with this section of the class.
Yeah.
What's your MH again?
MH, this is the total MH. It is, so these
Yep.
four corners.
No, what is MH? What does MH stand for? Sorry, that's what I was. I can't remember. That's
Mid homozygote.
Thank you.
Value.
So you think we need to go the harder route on this? We think A is correct to begin with. The very first question.
No, that's what
Nothing's
this is
right. Okay.
doing. Yeah.
All right. Nothing's right.
Yeah.
Well, that's not a bad point.
I'm pretty sure I calculated everything right too, like, because those four, and it's this minus that minus the average for A. Yeah. And for AB, it's the average. U of A is your mid minus 2 homozygote average. Same with that one. And this is the one where it's just like, eesh.
Oh, jeez, I lost you. There we go.
Oh, actually, you know what?
No, I don't. But
No,
what?
don't.
Tell me.
No. Oh, hang on. Okay, that's right.
Okay, I just need to box this stupid box in because otherwise I'm not going to know what the heck I'm doing here.
Okay, yeah, I thought for a second I mistyped in this equation it was going to be different, but I don't think I did.
And why are now? Maybe I'm dumb here, but why are you using 0.6 minus 0.4?
So it is the frequency of your big A allele minus basically the Q of big A. So whatever else.
Well, the reason I'm asking is that we don't have values of 0.6 and 0.4, do we?
Well, so big A, homozygous
Yep,
big A
yep,
is 0.36,
yep.
and then divide or squared is 0.6, and then same with the
Okay,
point
gotcha, gotcha. Okay. I wasn't thinking of squaring.
Yeah, then after this section in the notes, then it goes into epistasis, which is where you start taking your averages for loci and all that stuff. Sorry, did you guys need me to say clicked on that?
No, I was looking more at, let's see, your MH. That's just your four corners. That's where I was talking about earlier. Your four corners divided by 4, right?
Yep.
So add them all together.
Yeah.
Yeah, just run it through that. Yep, okay, that's right. And your A sub A, I can't remember, what was the calculation that you used? So that is, and I just want to see if I remember this now. that is, you're using C3, so that's the average number.
Yep.
Minus C3 plus G3. Now C3 you're using because of that, G3 because of the homozygotes. Got it. And then just by 2.
Yeah.
I gotta make sure I get all the stupid. Okay, I got that figured out. Look at you just go to town. Boom, boom, done. So how do you get them to subscript so quickly? You must know a command rather than...
No, I just went in the font and clicked subscript.
Okay, that's what I was doing too, but for some reason mine's a lot slower.
Oh, well, like, I just copied this down. Like, so I went like this, and I just went like, bam, bam. And then I went in and just changed the text, because it's easier that way.
That makes more sense. Okay, so... I gotta go check on my stove just a minute.
That's not right. That 144 right there, though, that's pretty close to the unweighted marginal mean that we were like looking at earlier.
Yeah.
So I guess then for let's see here. For average for the A locus, I'm just gonna like put this in there, I guess. And I'm gonna do same thing with B basically. Because there's not just like one for A, I don't think. You know, you have your separate ones for your different genotypes. At least going off the notes. I don't know.
Is that a purr?
Yeah, that's my cat. He needs attention.
Yeah, they say cats love quantitative statistics.
Oh, yes.
That's really one of their charming qualities. I think that's why we let them be a part of our domesticated lives.
Sounds likely.
Hey, just to check, how did you get your MH value? Is that just for the A or is that everything?
That's everything.
Everything, okay.
Yeah. I assume, do we have to use by letter later, I think. By letter later.
Maybe later, yeah.
By letter later. I don't think so.
No.
Wait. No, I don't see it. Maybe, I mean,
Okay.
might just not be
No,
seeing
maybe
it.
not.
It's already 7:30. God.
Happened so fast.
I still have to go to the gym tonight.
You go to the gym? I'm laughing because I haven't went in years.
Oh, yeah, I've been trying to. Well, I've been somewhat consistently going for a while. The problem is you hit harvest, and then I just eat like garbage, you know? You're in a flat combine for like 12 hours, and then you just crawl out and you'll eat the first piece of like disgusting gas station pizza that you find.
Casey's?
All right.
Yeah!
That's what I used to live off of.
Yeah. Yeah, so I'm from Ohio originally, and like they don't have Casey's out there. So I got out here and I was like, what is this?
Amazing. But terrible.
Yeah.
So much grease.
I also managed to move, so I lived in Maryland before living in Iowa. I moved 1,000 miles, and they built a Dunkin' Donuts like a month before I moved here, right down the street from my apartment. I was like, it's destiny.
Never had a Dunkin' Donut.
You should. I heard from a reliable Canadian friend that it's better than Tim Hortons these days.
Oh, Tim Hortons donuts are real bad, so I believe it. Sorry, Canada.
Yeah, he was telling me something like they sold their coffee recipe to McDonald's or something, and I was just like, this is crazy. I was like, how's this not national news?
It was. People were really upset, and that, yeah, like, they got bought out by Burger King and stuff, so, like,
Oh,
it's
maybe it was
like,
Burger
it's the
King.
most
Yeah.
Canadian thing ever. It's like, no, it's not. It's not at all anymore.
Let's see. Other means, Oh, I'm dumb. So, your average for the loci, like, you know, I did the average for these. You don't report these, you just take the average of those, and that's your loci. Sorry.
I'm playing catch up right now. I had a little soup break.
It's necessary. You gotta stay fueled.
That's right. Well, and it's the most appropriate thing to do when you're looking out the window and it's raining and then hailing and then snowing and the sun is shining.
Can't be right, because these things average out to the same thing. Hang on. Never mind. I don't. Because I don't think there's a single locus value. Is there? I'm gonna stick with this. I guess.
It's not too different than what we did before, except just that that average ends up being a different number because we have to get our MH values for each loci.
What, this one? What,
Yeah.
So, actually, Actually, what we more closely calculated last time, I don't even know if this gives a. Oh wait, it did. I said it earlier. What was it, like unweighted something? Do that?
Is weighted.
Hang on. I can't find it now.
All right, I'm back.
Welcome back.
And just to show my, my Canadian roots, I'm eating Kraft. Cassandra probably knows what that is in Chelsea.
Like the cheese.
Dinner?
Yeah, Kraft dinner. Macaroni and cheese.
KD.
So I take it everybody's feeling more confident in this new way of doing it?
No.
Oh!
Not really.
Uh-oh.
Huh. Let's see. All right. Here we go. Look at this. I'm gonna do a thing.
We're watching. Do a thing.
Non-epistatic.
That's all I have for this second. Let's see here. Yeah, so this is like just an easier table.
I did D and E, or yeah, that's how I was doing it.
Oh, you did these already?
Yeah.
Oh yeah, you guys said you went ahead, right?
Huh?
Because these are just ew. Yeah, so you get those. 14. Where the heck did we get that?
Question, what's your population average based off? Is that based off your mean heterozygote? Oh, that's the really long equation with A.
Yeah.
Based off the frequencies.
Yeah, so this MH is off the 4 heterozygotes.
Okay, and then that final equation is where you have to take in the 0.6 and 0.4 and everything.
Yep.
Okay, makes sense.
What's your DA based off of? For some reason, I can't get the same number.
So it's
Oh, the mid. Okay, that's it.
Yep.
No, don't mind me. Oops. All right.
That equation's off.
Woo! I did that wrong.
Oh my.
For the MH, I put the sum of all of them multiplied together divided by 4, and it's a very big number. It's wrong.
Oh, oops.
So for the non-epistatic value, it's just why do they do all this other equation?
So that you can say you did it?
Because it's literally like yeah, hang on.
Okay, what am I? H2 or H12 times 0.04.
Like, is that someone that did the epistatic already? Can you tell me if that's what you got for?
I'm not getting the right population average, same as yours, and I'm pretty certain that I've got the same equation that you do.
This one?
Pretty certain. Yeah, okay, M7 is there. I don't have exactly the same cells, so I've got to...
I got it.
No.
I wonder what happens if maybe I need to add some more.
Yeah, but I didn't do that either. I did it exactly the same way you did. M3, yep, okay.
Oh, sorry.
I mean, I'll put in the parentheses like you're doing there to make it a little more. H12 at blah 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 blah. H13, yeah, blah blah blah. But I'm not certain that that is. Okay, your M5 is.
My M5 is my age.
AB, and my AB is H13. I have in the same spot. Okay. Yeah, it looks like everything's. I'm getting 50.15. That is odd.
Don't know about that one.
Yeah, I got the same thing. The 127.35.
Yeah, that's why I'm saying there must. And I've got the 168, 116.5, and again, same values for everything else. What am I doing wrong? I'm wondering if I have a plus instead of a times or something like that.
Oh, give me 1 second. I can click back onto it. Sorry.
Yeah, don't worry about it. Just eating my craft.
Wait, why did I do that? How's that right? Right, add them, and then subtract your thing. Yep. Okay. Alright, sorry.
Yep, just put it there. You know, my. Oh, of course. Just a minute.
Wow, Jux is trolling me?
I think so. So these, like alpha and D values, I missed where these little guys came from.
Oh, yeah.
I tried a couple things and it was not happening.
So basically, the A's are your homozygous well, I'm going to say dominant because it's better than saying big A, little a. Homozygous
not troll you. No.
Minus the average of your homozygous dominant, homozygous recessive.
Yeah.
And then A of B is the same thing. And then D is just your heterozygote value minus the average of your homozygous dominant, homozygous recessive.
Okay.
Does that make sense?
Yes. So, such a tiny little screen. I need to make it bigger.
Oh, here. Yep, hang on, let's see.
And you're saying then that your population average is your 144.11?
No, that's unweighted.
Yeah.
Unweighted doodad average.
No, I meant your the average for the A and B locus.
So for my answers for those, I just put these listed individually because
Okay.
they're not. I mean, if you put the one average, like it's both. They're both going to be the same answer.
Mm-hmm.
That's why I just put these.
Okay.
Whether or not
That's kind of what I was wondering.
I'm just curious, like, if those are the average for each of those genotypes, then what are the 150, 163, 383?
That's average cyst counts for those loci.
Right, okay, and you're saying average genotypic value.
Yeah, so in the notes on page 2, and it says this under single locus too, so keep that in mind. I don't know. I mean, this is the only thing that's like made sense to me to actually get some of these values and everything. It says to draw inferences from the genetic properties of a trait, we model phenotypic values using linear components. Most common model, blah blah blah.
Found it.
P equals G plus E, where P is phenotypic, G is genotypic, and E is non-genetic component. So if we assume that the sigma E equals 0, or the change in E is 0 in between all your entries, then sigma P equals sigma G, or absolute P equals absolute G. So the only thing that we can do from this information we're given, because these are all phenotypic values technically, right? Is assume that our sigma E is 0. If that makes sense. Because otherwise, it's just like this data set, and we're like, well, that sucks.
Does it really suck? I'm not sure.
I mean, if we could do all these calculations, how much fun would that be? Not a lot. TBH.
You know, I could use this time for something else, like not screwing up my Excel calculations like I just did. By the way, I put a minus sign in instead of a time sign in instead of a minus sign. That's all it was. I trusted you.
Whoa, that's not right. What am I doing? But it is right.
B4, P8.
Oh, yeah, that's not right. That's better. Just let me know if you need me to click back onto anything.
So far, everything's matching up now that I've got all my correct. Uh-oh.
I thought I saw what was going on, but I did not. Yeah, okay, I got one of them. My brain was having a little meltdown doing the back and forth between the spreadsheets because I don't have mine set up exactly like yours. So I was like,
yeah.
yeah, and then this
Yeah, I'm just going back through and doing them like yours because that makes it a lot easier to find some of this stuff.
I'm honored.
Well, I have been treating these past 2 weeks as a great opportunity to learn about better organization.
You are looking at the wrong person's stuff then.
Yeah, well, it's still better than the way that I've been doing stuff.
I organize with the piles method. You ever tried it?
Yeah, actually, that's kind of what my life is all. Why does it keep changing? Oh, I put capital A, small a in, and it keeps going, no, that's got to be a small a.
It doesn't like that.
It really does not like that. What is wrong with you?
So you guys had said you did D and E already. Are these the values that you were coming up with?
It looked like it last I.
Seriously, I can't get it to not do that. Oh, that ticks me off. What in the heck is going on?
Yeah, it all matches.
Cool.
Okay, what am I doing wrong? I'm serious. I cannot get a G capital A small a to work. It automatically just turns it to G small a small a.
So they,
Every time.
that's
Even
weird.
if I change the cell formatting and everything.
Have you tried highlighting the 2 letters and then go to font and then do your changes?
No, it's not that. I'm literally typing in just into an empty cell, any cell, capital G, capital A, small a. I do a space, and it automatically goes G, small a, small a.
Capital G, big A, little a.
Mm-hmm. Yeah, see?
It does, doesn't it? That's annoying.
Yeah, I don't know how in the world.
Like I just did. You want You want to do that?
Yeah.
Just hit Control Z, man. Oh, sorry, wait. No, Mac has Control, right?
I'm not using a Mac. I'm at home.
Oh, gotcha. Yeah.
Hey, wait.
So here. So G capital A little a, right? And you hit space.
Yeah.
You changes it. You just hit Control Z.
Thank you.
No problem.
That helped a lot.
Okay, so I got the alpha ones, and then the DA and the DB were a little bit different, right? Because they're not the homozygotes, they're the heterozygotes.
Yep. So it's your heterozygote minus, and then still the average of your two homozygotes.
Oh, okay, so it's pretty much the same.
Yeah, it's basically the same.
Okay.
I'm also impressed with the whole merge and center thing that you're doing. I mean, it does make it look nicer.
Oh, thanks. I minored in Excel tables actually.
That's what I'm saying. This is where I need the work, and you're
And then
definitely helping. I excel in the just place it over there, and oh, by the way, that's a mountain of stuff. I'm not quite as bad as a hoarder, but I've been accused of it. How about that?
Yeah, that's not great. Oh, I see. I see here. So the because that's the next one, right? Epistatic, yeah. So it's just the difference.
Yeah, so, I mean, really, shouldn't we just prove that it's either epistatic or non-epistatic?
Well, I thought we were calculating.
Yeah, that's what I was assuming.
Are we proving one way or the other?
It says calculate B and then it lists everything.
Yeah, because after you get the non-epistatic values, then you scroll, you just keep following the notes. I mean, something can, like a gene could have an epistatic effect and a non-epistatic effect,
Yeah, no, I think we calculate both. I was just looking at the one example in the lesson when I was doing it, and like he had to change a value for like the 1A. As long as it comes out 0, then you don't really have epistatic. But maybe this
So-
situation does have both.
For your non-epistatic value, what's your formula for that? C21.
C21? Okay.
Yep.
So it's your,
Yep. Oh, I haven't calculated that. That's why I couldn't find my value. Okay.
oh yeah. Those are just the averages of your,
Yep. Yep.
whatever this
I had asked you about it earlier, and then I didn't even do it myself. Yeah, same thing. God, that makes sense too.
Sweet berry wine. Oh, you know what? That doesn't work. That drag doesn't work. That sucks.
And there it is.
Hey, it looks like we did the next part correct when we did ALA 13.
Yay!
Oh,
You remember LA 13?
Well, I knew we'd done like some of this before, and I was just looking up the graded document.
Oh, I gotcha.
I'm still fighting with question 1. For the recessives, I keep getting minus 13, and I just have the inverse of that when I do the 2 of them, so I'm doing something wrong.
Wait, for which one of these?
Like the MH values for heterozygote A and B.
Oh, so I didn't do the individual ones. Because I don't think you actually need to calculate that for any of the stuff we're doing.
For column L, 6 and 7. And then over column M.
Say that one more time?
So the little d subscript A.
Oh, you're doing these two?
Yeah. Yeah.
Gotcha. Yeah,
Oh, I see what it did, I think. E5 and then the heterozygotes added together.
or the homozygotes, you mean?
Yeah, the homozygotes. I was doing the heterozygotes.
Yeah.
And I was just getting like a weird inverse relationship number thing.
Yeah.
And it was weird and confusing, but yeah, that makes sense. I see what I did. Thank you. Now I need to figure out the crazy equation.
I don't like merge and center for some of this stuff because it doesn't let you drag.
Yeah, I noticed that too. But you know,
What you gonna do, right?
sometimes you just gotta live with it.
You have to suffer for your beauty, right?
Yeah, exactly.
I was gonna say something along the lines of like, style is suffering.
Yeah, pretty much. That's a good French thing.
Hey, this goes back to what I was thinking when we first started doing this, because I knew these numbers, these recessive ones, would have to end up negative at some point.
And it's all working out.
Whoa, what the? Why is that AA? Oh man, that's probably like that on my. Yeah.
Whoa, I made that so big that it just like turned invisible.
Minus that. I should probably make sure I'm actually subtracting these the right way.
Oh, whatever. All right, non-epistatic matches you. Always a good thing.
I mean, unless what I did was completely wrong, like the first time around.
Well, no, it makes sense now that I thought about it. I just didn't expect it at this point, I guess.
Yeah.
Sorry?
What was that?
I don't really know. Kind of like a hiccup, kind of like a burp. Not quite rude, but hard to ignore.
So for your epistatic values, how are you formulating those?
It's just this minus that.
I didn't even see that, sir. No, not your epistatic. Epistatic. There you go.
Yeah.
Okay. Because it's highlighting you. I've got. Oh, sorry. I was looking at your yellow cells, your H and your 21. I'm like, how the hell is that?
Oh.
No.
Yeah.
Hot damn!
For whatever reason, it the boxes, your highlights around your boxes are not very highlighted.
Yeah, it's kind of hard to see.
All I'm saying.
It might have been because I zoomed in, so it's like not as.
No, it's okay. I was just wondering. Now I know.
Right, I got the same non-epistatic and epistatic values.
Swag.
So what about for large A, large B, small b, the 1.1111? So I understand how you got the between the two large, but what did you use?
I'm sorry, I zoned out for a second. What was it?
Highlight H22. That'd probably be the better answer. There we go. Okay, so you just went down for BB there and BB there, so it's just a matter of that in the AAA column. Okay.
Sorry to backpedal. For B and C, for the average of the A and B locus then, is that like the 78, 122, and 231, and then 75, 124, 132?
Yep.
Okay, I'm just gonna change where I put this in my document then.
Yeah, that's what I put in. I mean, I don't know if that's necessarily right, but there's nothing that I can find that makes me say, yeah, this should be something else.
Cool. Alright, so now I need to calculate my epistatic, or my D and E, because we do them together.
Yeah, basically.
Yeah, everything I've got matches up with you up to this point. Most excellent.
Cool. And that's I'm stopping because I'm at 62s. So.
Listen to you. You're so funny.
So funny.
No, I'll stay on for like 5 more minutes, but then I should probably go to the gym because I'm waiting to eat until after that. I'm starting to get hungry.
Fair enough. So we've got question 1D done. Or is it E?
Or through E.
E. Gotcha. I'm labeling now. That's how scary this is.
Average allelic effects. I don't think, oh, here we go. Maybe not.
Here we go.
Happy static.
Problems?
That on page 7?
The next part, F?
Yeah.
Yeah.
Oh, yeah, I just kept like reading towards the end. I was like, why isn't it further?
Are you going to get on tomorrow night, Kyle, or are you going to just do this all on your own?
I'll probably be on Thursday night. Tomorrow night, I'm dedicated to playing a children's party game with one of my friends.
That sounds kind of cool.
Children's party game.
Yeah, I got a Super Smash Brothers tournaments
Oh.
So why do you call it a children's party game?
because it's like a long. Oh, it's not that long. So, like other fighting games don't like the Smash Brothers games as much because they're not like exactly the same. So me being a smartass, I made up a really long genre description for what Super Smash Brothers is, and it starts with children's party game.
Oh, okay, cool. My son would probably be kind of pissed at you that it's called that, but other than that, okay.
Nah, so you just gotta own it. It's actually really funny when you get a bunch of people to own it. It's like, yeah, we just play this children's party game.
Well, I actually play D&D.
Yeah, I've played a couple sessions. It's super fun.
Yeah, like 2nd edition, like, real old school.
There you go. I think I played 3.5.
So, I kind of feel your pain. I also play World of Tanks when I'm not in graduate school.
Yeah, I've heard a bunch of people play that. It's pretty fun.
It's a dangerous thing. It sucks you in. Anyways.
You're gonna be able to have so many hobbies, Jacques.
Well, it's what I used to do before I went back to school. All right, average allelic effects for each locus. That moves on to
Page 7.
Sorry. Again.
well, I was going to say that means that we move on to a different data set, correct?
No.
Oh, no. We still have that. You're right.
Yep. Man, maybe I really need to go to the gym, but I kind of want to stay on and do this because I know how I get when I stop doing an assignment. I'm like, I don't need
Well,
to.
jump back on later tonight. I gotta go. I actually have to jump off here for about a half an hour, 45 minutes, and go pick up my wife and the guys from the track meet. So I will be back in by 8:30, I'd say.
Actually, yeah, I might do that. I probably won't be back on for another hour, though.
Yeah, somebody will be here.
Sick.
So see you guys in a little while.
Yeah.
See you guys.
See ya. What page are the epistasis values on? I'm behind. I stopped to eat soup and I'm paying the price forever for the rest of my life.
It was towards the end. I don't really pay attention on
Like, I see the 10.
17, I think.
Okay, yeah, so I'm like right looking at the table.
Yeah, I can pull mine up if you want to see.
Yeah, okay. Like, yeah, I could see the values that Kyle had, but not the
Yeah.
equations, and I
So
don't
you-
want to kill
Sure,
him.
so you already did the non-epistatic?
No, I don't
No?
have that
Okay.
either. I just have two
So-
bulletins ready to be filled.
Okay, so the non-epistatic is just comparing to the frequencies. So you're taking, basically for AABB, you multiply, or you add both of those frequencies and then subtract the overall average.
Okay, and I'm taking the average frequency, so like 75 plus 78.3 repeating.
Yep.
Divided by,
Minus.
and I'm subtracting it from
The overall average, so 144.
144.
Yep.
And the same combination over and over.
Yeah,
except
just
that-
adjust to whatever the actual frequencies are.
Perfect. And then,
And then
Oh,
epistatic
yeah.
is just taking this value, your original, subtracting the non-epistatic, and getting your new to show what the value is.
so it should be 21, whatever.
Yeah.
Cool. I'm going to tick along on those and then listen to you all moving forward.
Alright, I gotta run and I have, I'm chaperoning my son's field trip to the Bay Island, so I will be offline till Friday night.
Yeah.
Okay.
You had this field trip right during the final.
Yeah, it's perfect timing.
We'll have
So yeah,
some
I'll
field trip.
watch and I'll probably be on Saturday morning.
Alright, sounds good.
Talk to you later, have fun.
You too.
Always. What the heck am I doing? So if it's a heterozygote, you just don't do the math, right? Because like for the homozygote, you take the average of big and small.
You do that same calculation for everything. So like, because you're talking for the big A, little a, big B, big B. Is that what you're on?
I'm looking at the grid the wrong way.
Okay.
I see what I'm doing. So yeah, now it would be, if I move to the right, then it would be big A, little a plus big B, big B.
Yeah.
I just having a thought. It just came to my mind. I don't know why, but just because we were talking before about how it also says, like for going back to number 1, how it also says that the sum of the genotypic value times genotypic frequency also equals the population average, but they don't equal the same thing in our case. And I was just thinking, I wonder if that's because there actually is environmental the residual value or environmental value or whatever it was that we decided would be 0. Because we had to assume it was 0 to make that equation work.
In this assignment, or are you going back to the?
No, this assignment.
Okay.
for like the population average. Because
Yeah, I
you can use that equation, but you can also, it says population average value can also be calculated as the sum of the genotypic value times the genotypic frequency, which is what we originally did. So my question is are those values different because there is actually the E or whatever? What did they say it was called? The environmental effect?
I don't know, but that sounds like it would make sense because, like, you'd think that, well, I guess we have multiple data sets to look at, but, like, you think we would be considering.
I'm just wondering that because I thought Kyle said that you have to assume that the environmental effect is 0, which just seems weird to me that we would be doing that.
It's a weird assumption to have to make.
And I just had that thought about like maybe that's why those two numbers end up being different. Because they're not like super different, but they might be different enough like for environmental effect to be the difference.
And you're talking just comparing the 127.35 to the 144? Or, because like,
48 or whatever.
the 148? Okay.
Yeah. Well, just because, yeah.
Yeah, because the 148 and the 144 were pretty close, but now the population average we calculated was like the 127, right?
Oh, never mind. Because it just says that you assume the phenotypic value is equal to the genotypic value. So you'd still have to assume that the phenotypic value and genotypic value are equal, and so that the environmental effect is 0.
Okay.
I was just trying to figure out why they end up being different because they're supposed to both equal the population average.
Yeah, I get what you're saying. Not sure.
It's just, I don't know, it just bothers me because I feel like what we originally did should come up with the population mean.
Yeah, I don't know. Maybe it's one we could post on discussion. Because, yeah, right now I kept both in my Excel sheet. Kept the old formula. I mean, I feel like I get where this one's coming from.
Yeah, so do I totally. And I just can't.
It's just which it is.
I just can't figure out how the other one would be wrong.
Yeah.
And I agree that I see where this one is coming from as well.
Well, and like in the grand scheme of things, that like they were off by 20, and I don't know how to interpret that value. You'd be like, oh, 20 is a lot, or not 20 is negligible. Dare I say I don't know how significant that difference is?
I'd say it probably has to be like fairly significant because the range of the averages is what, like it's 31,377. So I still feel like a number of 20 is still like a significant number.
Has anybody calculated the average effect of the allelic effects for each locus?
I'm working on it.
Okay, because I got mine, but I'm not sure if it's right.
Okay, let me finish one last equation and then we can compare. Okay, do you want to share or do you want me to share?
I don't. I guess I can. Mine hopefully isn't too much of a mess at the moment. So this is what I have. Oh wait, I think that's supposed to be A, and that's supposed to be little a. But they're very high. So I'm wondering if it's supposed to be done differently.
Yeah, I got different values. So I've got the 0.6 and the 0.4 that you had. Here, I'll share mine. So this is how I did it. So I think we had the frequencies the same, right? 0.6, 0.4.
Yeah.
And then these were the equations that I used for A and D. And then these were the equations that I was using for the allele effect.
Yeah, I was using, I think, the same. I had the same allele effect equation.
These ones?
Yeah.
Okay, did you have this?
No, that was.
Okay, so that is on page 5, is where I got that.
Okay. Yeah, I was just following what I had done in ALA 13.
Oh, okay.
That's why I didn't have much confidence in it.
Okay, I hadn't gone back to 13 completely. Probably would be good to look at that though. Let me go back and look at that.
And I'm back.
Hello.
Hello. Have you guys gotten past E?
I was working on E, or what's E?
E's the epistatic values.
Oh, yeah.
You guys are in F.
Vortex.
I'm trying to remember which ALA that was in.
Oh, it's 13.
Oh, I was just one in front of it. Okay. Okay, yep.
Yeah, because looking back at 13, like, I didn't have the population means right. What's, I got a 17 on 13.
So did I. But
Yeah,
he
so did I.
didn't mark anything wrong with that problem, though.
Which one?
Like the question about the allelic effects, he didn't mark anything off on that, I don't think. Not on mine.
Yeah, I don't see any. There's nothing marked on mine there either.
Yeah, but to get the average effect, don't we have to give it each allele? I suppose that's what our alphas and our Ds are, but our A's.
You calculate different ones, I think. Is what I was looking at on page something. Find it again. Page 5 is where I started.
I have no comments on Mike's ALA 13 either, and it was returned as a PDF.
Oh.
So I don't know if I was right or wrong, but I'm assuming I did the same thing you guys did.
So I'm gonna pull mine up again and let's see what we did differently, Emily.
Okay.
I think Chelsea or Michelle. Michelle. Well, whoever had done the average. Sorry, I don't mean to be cryptic here. So whoever, I think it must be Chelsea then that did it similar to me, as far as for how it got turned in. So we had the M1TC locus genotype, and we did a frequency mean, and what we should have done was the theoretical mean. And that's where, so I'm saying that otherwise everything apparently was correct, because if I had done that in my answers, then he would not have gotten after me. So anyway.
Which question are you talking about?
ALA 13, number 1.
I apparently got it right.
Yeah, that's why I'm saying that.
I just don't know remember what it was. Number 1?
Mm-hmm. So you have your tables, and then you calculated the theoretical mean, correct? My point of it wasn't so much that that's what we need to be looking at. I'm just saying that all the calculations we did to get to that point apparently were correct. I just don't understand how to give the genotypic value to our loci for our question.
So which question are you on the test?
On the test, I'm on F, but I thought that had to do with the genotypic value to calculate our.
I thought we were making the assumption that the genotypic value is equal to the phenotypic value.
Okay, I wasn't here for that conversation. Okay.
Like that was the decision made when we decided to calculate the population average in a way that we did, because that assumption had to be made.
Okay, that may have also been when I wasn't on. Meaning I was kind of on and off here for a while.
Okay.
It's been a weird night.
Okay.
Meaning I haven't been devoted, not like you guys. Anyway, so we're going to go with the phenotypic value is the genotypic value. Okay.
That was my impression anyhow. That's what
Sounds
I
good.
gathered.
No, I'm not arguing. Let's do it.
No, I'm just saying that I don't promise that I'm right ever.
Our confidence in our abilities is shocking, I know.
Okay.
Okay. So I guess I had calculated the P and Q frequencies, and we had the same there, right?
Yeah.
Okay. And then, so this was what you didn't have, right?
Yeah, I did not do that.
Okay, so I had done that. So that was what I said was on page 5. Because on page 7, where he's calculating it, he said he referenced the table from 4.2, and that's where like he calculated like the 8 and the 4 values. So that's why I went back to page 5 in the notes and used those equations to calculate A and D.
Okay, I just went by that table 4.3. So I got A, P, Q, and D from that table.
So you just use this as A directly?
No, it's a different table on page 7.
Mm-hmm.
Well, so wait, what did I use as A?
Because yeah, I basically calculated my A value as this plus this divided by 2.
Mm-hmm.
Okay, yeah, and that's what we did to get our values for question 1, kind of.
No, we were getting the A sub, the small A sub large A, whatever.
Yeah.
The MH and then the list of stuff that she has there, which is.
Oh, yeah.
Oh, I'm sorry, Michelle. I gotta say, I'm sorry. I thought I was looking at your ALA 13, and I'm like, that's a lot different than my. No, that's all your test stuff.
Yeah,
Oh, sorry. So, yeah, I was gonna say that looks really good. Wait a minute, why are all the numbers exactly the same? That's pretty okay. That's you are quite a ways. You're quite a ways along. Okay.
So, Emily, what were you using as your A and D value then?
So I had the scale 4.3 at the top. You have, I mean, I guess it's similar, but yeah, the genotype and then the coded genotypic value is what I was using as A and D, and then the frequencies, of course, were P and Q.
But what are you using? Like, what's your coded genotypic value?
I guess I did just take the.
You just use these.
I need to get back to that. Yeah.
Because, like, when he referenced table 4.2, he's got a genotypic value, and then the coded genotypic value is different.
Let me.
Now here's a question for you, Michelle. For D, should that be YAA plus YAA divided by 2? Should that all be in parentheses first and then minus that from Y?
Yeah, it is in my equation.
Okay, just making sure. I thought so.
Yeah, I'm pretty sure, yep.
So for the alpha,
So I guess does it make sense to use that coded GV and calculating it the way I did, or is that not? Do you think that's not for this situation?
No, I think it makes sense to get that.
Okay.
think it was just done differently on our last assignment.
Yeah, and since I got things wrong, I'm not 100% confident.
Where are you getting the stupid single screen? Where are you getting the equation for A and B? What page did you say?
Page 5.
Oh, duh, I went right by it. Okay. Okay, yeah, easy enough.
Okay.
So how did you calculate, just, I want to go back and double check on this, your frequency for P, capital A and P, or Q, small a. So you have your 0.6 and 0.4. Just wondering how you,
I did
0.48 times
C2 plus half of D2.
0.5, you use that, okay.
Yeah.
Okay.
And then same thing for this, just half of.
I know I should know that, but that seems too simple.
My brain is not functioning tonight.
I'm gonna play with a couple things here.
I don't know if my brain's ever gonna function again. Not to try and one up you.
Yeah. Can you click on 0.6 again? Sorry. On your P. What am I doing wrong?
So there it is.
Why the heck? Okay, I'm really kind of getting tired of, instead of just putting 0.6 in there, I'm trying to do exactly what you're doing.
Mm-hmm.
And for whatever reason,
Did you create your own table or copy it from his thing?
I created my own table.
Okay. So now he had random, like, text box and shit in there.
Okay, let me try. Okay, I got it. It's just, I had one cell wrong. Man, Excel is just tweaking me off tonight.
Did it let you use the square bracket in your equation?
No. I just had to do extra parentheses.
Yeah, it yelled at me too. I was like, fine.
There, I got it. Oh, some days, I swear.
It's negative. Q times
Okay, are you good if I stop sharing?
Yeah, I'm good.
Okay.
Although I didn't copy any of what you've been doing down. I'm trying to catch up.
So for your average effect of the allele for A, big A or dominant A, did you get 115? Recessive 172.32?
Yeah.
Okay, and then
And my 172 was negative. Was yours?
no, but I feel like it should be.
I had it negative because the equation was negative P, I think.
Yeah, that's right.
And which page are you pulling that from?
5 and 7.
Okay.
And then B was 151 and negative 64.83.
Yeah.
Okay.
I guess my 151 was 0.27. Did you have that? I don't know why I have decimal places.
Yes, I do.
Okay. Okay, perfect.
I'm gonna move that down. All right, Michelle, could you share for a moment?
Sure.
I've got.
Ignore this part. I'm still playing with this.
No, I just got my allelic frequencies figured out. I'm gonna change mine to match your. Oh, okay. I see. Oh, I see what I did. What? Why in the heck is that? For some reason, you made M. Oh no, it's not a. It's not a. Oh, you're working on your stuff. I was going to say it's not a. It's a formatting thing for the cell, so that makes sense. That and going down to your formula, I wasn't using. Why are you using brackets instead of quotes?
I wasn't. I just wrote out the equation the way it was in the notes. But
Oh, okay.
in this, I did it the same way. Like parentheses, it will make you.
Right. Well, that's why I was just sitting there going, that doesn't
Just to
match up with
confuse
what
you.
I'm okay with that. I was looking at the way it was written, and I'm like, okay, but I haven't been using brackets. What does that mean? Because who knows? Maybe it's stupid that way.
Yeah.
Yeah, like you guys, I'm like, oh, did I do that right? Did I do that? Oh, did I? Yeah, maybe. No.
I just keep getting a reference error.
Gesundheit.
Thanks.
Reference error on what?
For little a, big A, the one with the first one that you had there with square brackets.
This guy?
Oh, you're sharing again.
Yeah.
Yeah, that guy. Okay, what am I doing? What am I doing? And, okay, yeah, I did add a times, and then I took it out by clicking, and a bracket, plus, and a that, and I need one more bracket. Okay, I got one more.
That's the sound effects alone are worth this tonight. I'm just trying to figure out how I screwed up my. How did I? What in the world? Oh, got it. My problem is that I like to organize things, and I like how you have frequency and then 0.6, 0.4, and your numbers all the way down. And I had frame shifted after my P and Q, and I'm like, what in the world? Well, now I figured it out. You have a big M box.
A big M box?
Your column is wider, so mine was tiny, and it was.
Gotcha.
Yeah, I screwed that up.
Gotcha.
I hear you, Michelle. I wasn't 100% sure what he was referring to.
Oh, jeez.
Well, no, it wasn't a very precise explanation.
Yeah.
It's like, oh, you did this thing with the thinger. It's like, what thinger? There's lots of thingers. Do you think a thinger is what I think a thinger is?
Got it. Yeah, I feel dumb. Anyway, that's okay.
I feel dumb all the time. I do this all night. I feel dumb. I go to work. I feel dumb.
Really?
Yeah,
I would imagine that you are not one of the dumb people at work.
I'm not, but that's why I think I'm dumb, is because I am extraordinarily hard on myself.
Yeah, my wife says that about me too.
And unfortunately, no one gives you prizes for that.
For being hard on yourself? Yeah, no.
Yeah. Well, I was telling my breeder, I was like, oh, how am I supposed to cheerlead other people when I just, like, feel so defeated? And he's like, I don't know, I can't help you. I was like, oh, good.
So this part I'm not sure on.
Oh, what did I
So whenever you guys get to that,
Yeah,
let's discuss before.
Can you go to your small a, large A, 114.88? I feel like I'm missing or put a parentheses in wrong. Okay, so you're referencing that, right? Oh, that's what I did wrong. Let's see if it works now. And I didn't do that. I keep forgetting that you have to put those times symbols in. Okay, so, and your answer was, you can unclick 114, mine's 98. Yeah, I got something else. I bet I'm missing a parentheses. P4, blah, blah, blah. Okay. So what am I missing? Okay, I'm just going to copy your parentheses setup and see if that matches. Nope, that didn't change it. So what value do I have that's off from yours? 266 minus 103.6.4. It should all work out. Is it going to be this all night where I'm having these issues?
What did you say you were subtracting?
Okay. I'm going to look P4. Like I said, mine's off a little bit. So I'm subtracting. I can't look at your thing and run it. I've got
Sure,
Q,
so just,
yeah.
so is it pretty much, you want 0.4
Yep, I've got that. I've got that as
times
P7.
Okay, well then
It matches everything.
66.
Colors and is that right? Color wise, P da da da. Oh, wait a minute.
Was it a negative?
No, I think. Why did it do that? It looks like what happens is I switched my P and Q around in the last.
Oh.
What the heck? N4, which is, yeah, 0.4, right? Yeah, minus Q minus P. That's what I thought I had. Now it matches. 114?
Mm-hmm.
Huh. I was following the formula. I wonder why I... Oh, well.
I have different values than you for the average effect of the allele substitution.
Yeah, I'm not sure I'm doing that right.
I mean, from what we had last time, you just took, like, for this to be, like, the dominant minus recessive for each locus.
So you're just using these values?
Yeah. So I've got 287.2 and 216.1, which is a little bit different. But then again, maybe we're supposed to do it differently now. And I'm just being lazy.
So that's what you got? 287.2, 216.1?
I think so. Yes.
Okay, because what I was looking at was on page 7, where it has average effects of each allele can be calculated as. He had, like, the alpha of big A equals Q times your alpha value. So it was like 0.2 times 5.6. And then at the end, and he did that for the alpha small a, and then he subtracted that from that. So that's why I had done this, because that was the equation that I was looking at there. But I agree, because in the section above that, you do have just these values being your alpha, big A, alpha, little a.
Yeah, well then, yeah, I think I'm just gonna leave it because that's what because it just says it's the difference between the average effects of each allele. So.
No, I think I'm good with that. Because, yeah, I was not at all sure on this.
Well, and if you look at, like, the calculations, so he got, like, 1.12 and negative 4.48, and if you go to the...
Oh, yeah, they're the same thing. Yep, you're right.
Okay. Yay, we got another one done.
Which I guess is why this and this match this and this. Maybe?
Oh, yeah, that makes sense.
Is that right? Except that
some nights, I swear.
because these are allelic effects, right?
That we did an F?
Yeah.
Yeah. Isn't it?
And yeah, it's curious that like the way I calculated this for that average effect equals the A value, not the. I would have thought it would have been one of these, but I don't know. I think it makes sense just to do this because this would be our A and our big A, little a.
Yeah.
I was just debating if we needed to do this step, but I think you're right. I think we can get by without it because we're saying we calculated these two and it should just be the difference. I need to play with my symbols. Sorry if that drives anybody nuts. I'm not that.
Oh no.
They're your symbols to do with that.
I'll get them in there at some point, I suppose. It's just a pain.
I don't even bother with them in Excel because they frustrate me.
Yeah.
Maybe I'm the lazy one.
I think that's what she's saying without saying it.
I think so. Q minus B.
Why in the world do I have that?
What I do.
You don't even know what I'm talking about. I know. I don't know what I'm talking about either. That makes... I have something that says YAZ instead of Y large A small a. No clue. All right. I will say this, Michelle, I appear to have the same numbers as you. However,
Excellent.
my sheet is not quite as well organized as yours, or pretty. But
Yeah,
I'm working
it's all
on that. I can work on that now. So we have allelic effects and allelic substitution. And what page was the allelic substitution?
7, I think.
7. Not on that one. Which handout is it? Sorry.
Quantitative genetic models.
Okay. Just want to double check.
My numbers are matching up too so far for the allelic effects.
Awesome.
So I can't disagree with your formulas for either the average allelic effects or the effect of a substitution, so that's a good sign, right?
Yeah. Well, that means I'm interpreting it right.
If I'm right, I have no confidence in my thing either, so. So we're all a bunch of chickens.
Pretty much, yeah.
I can live with myself that way. I'm not afraid to say it.
Whoa.
And okay, big reminder, save your work. This would be that time.
Now I want to do this all over again just to make sure I really know what I'm doing.
I seriously might throw up if I did that. Oh, I don't need that anymore. Let's see. There we go. So, how did you? Oh, you just added. Did you add another line in there? Don't. I'm just talking to myself here. So. Oh, you're putting in friggin' alpha symbols now. I know you were just talking about that, but oh, that's not fair. I am not doing that.
Yeah, you'll be alright.
Oh, I know I'll be
I don't
all
think
right,
so.
but talk about getting one up. Ugh.
Sorry.
No, everybody does what they need to do. Just look at you go. All right, allele effects. Age, focus. Allelic substitution. That's what I was working on. Is that? Hey, I don't know. I don't want to copy nothing. Jeez. What I'm going to do is. Just. Yeah, you do good organizing. I like how you're putting. I mean, you're putting. I was just putting like question 1, E, and F, and you're putting average effective allele. I like that better. That looks more professional. I'm debating. No, I'm not going to go and. It's tempting, but. Just copying you as far as how you're doing your labeling is enough. Oh crap, I really want to do it though. Insert symbol. If it's near the top, I'll do it. It is. I'm a nerd.
So has anybody started on H or I? The additive genetic variance, the dominance?
Yeah.
Are you still looking at like the dominance deviation in this same handout, or are you looking at anything else?
It's on the variance, covariance, and heritability handout on page 5.
Aha! Okay, are you good if I stop sharing?
Yes, I am. Thank you.
Yeah, probably.
So for allele substitution, you guys said that the single A and the single B is the same as what it is when we just calculate it as an allelic effect. For the substitution, effective allele substitution.
So we did like the difference between these two, the allelic effects.
Oh, okay.
Mm-hmm.
Alpha big A minus alpha small a.
Yeah.
Mm-hmm. All right. What have I not answered yet? I have not done G. Because I was making it pretty. Well, that's not a bad thing. Now I've come to appreciate, Michelle, that putting the formulas in is a good idea, and he seems to like that too.
Yeah. At least you can see where I'm wrong if I'm wrong.
Okay, so additive genetic variance, you said, is in which one? Which handout?
The genetic variance, covariance, and heritability.
All right, got it. I'm using printed copies here, so.
Yeah, I have them too.
Okay, so additive genetic variance. I remember this. We're back into, after a bit of algebra.
Hey guys, just wanted to talk about how much I like algebra.
Total genetic variance, we want additive. Additive variance, not additive, genetic variance. Oh, this should not be that hard. Where did it go? Have you found it?
Page 5.
Everything's on page
Emily
5.
found it.
Okay, on page 5, the variances. Oh, there it is. V add. So my question is it 2PQ alpha squared, or is it the one below it? Because he's got that 2PQ alpha squared highlighted.
Well, I think
Oh, it's in the
that's-
middle of it, so. Sorry, go ahead.
That's just a long way of writing that out.
Yeah, so it should be the same thing.
I'm having issues calculating it. I see an error. I think it's because of the negative.
Oh.
Which formula are you using?
I'm using a long one.
The 4P2Q2 alpha 2?
No. I'm using the 2PQ and then
Alpha D plus D?
that's, yeah.
The one right before V dominance.
Yeah.
Okay, well, I'll give it a shot and see what happens with me. If I start screaming, all right.
Okay.
Page 5 in quantitative genetic models?
No. Genetic variance, covariance, and heritability.
Oh. Thank you.
Okay, let's give it a shot.
Yeah, my first attempt I got an error too.
I think it's because of the negatives.
Give me a sec, I'm coming. Okay. Now, what do you mean the negatives? The only negative I see is Q minus P.
Well, D's negative.
Okay, you're right. Don't mind me. Wasn't even
Whoa, that is
there.
a large.
And then,
Where D is not in that equation though, right?
okay,
Yeah.
I didn't get an error, but I got a really wonky number. So what, Michelle? What? Oh, you're not sharing. I will share my really. So see my wonky number,
Oh.
but it's not 100% set up right. Should
Wait a second. How did?
Ooh, that's even worse.
That looks the exact same as before.
Wait. Are you using the last notes? The quantitative genetic model? Because I don't have the D. Am I looking at something different? I am looking at something different. Never mind. Because you're using the final one, the 2PQ A plus D Q minus P squared.
Yep, unless something. Well, that's.
Because I was using the bigger equation still.
Well, my question is this: why doesn't the 2PQ alpha squared work? We have alpha, and we have PQ, and obviously we have the number 2. So. So my only question is, oh, yeah, if D equals 0,
Which ours doesn't.
V add, is that, yeah, the final one, V add is 8PQ cubed alpha squared. That's if D equals alpha, which it doesn't. Okay,
Did we calculate alpha?
yeah, that's the average effect of an allelic substitution. I mean, that's alpha, alpha equals. Or no? Am I wrong?
I was thinking your alpha is P, the 266.5 value. And P. Right.
Yeah, okay.
That's A though, not.
Yeah, that's A. That's not alpha. That's my A.
Oh, you're right.
So,
So alpha is
what you have.
287.2.
Yeah.
Yeah, that's right.
I'm not saying I'm right. I'm just going, I put in that little alpha there because you did it and it looked good.
No, yeah, you're right.
So I'm just wondering if 2PQE squared isn't right. Or no, 2PQ alpha squared. And then you do that, and that, and that. And look what I get, the same thing.
That just seems.
Oh, it seems all kinds of wrong.
Yeah.
But that is following the formula as far as I can see.
Like all of our numbers for every calculation is pretty high compared to what.
But he was working with much lower numbers the last time.
Yeah, that's what I was
Yeah,
trying.
I don't know if we're out to lunch, but
That seems so high.
Yeah, that number doesn't seem realistic.
Insanely high.
no, we've got a lot of additive. This is an awesome crop. We have potential.
Wait.
You make these two things together, they're just gonna explode. There's so much added. Plant literally explodes because it's just overflowing.
No, I was going to say, you take the parents and the closer you get them together, the more they vibrate. No, boy, this could be bad.
My number came up even higher than your number. How did that happen?
I am the same as Shock.
Okay, how did you do yours?
Hi, Kyle.
I
Come on in.
P times P times Q.
We're having fun making big
Oh, I don't have
numbers.
Q in there. Why is my Q
Want
in
to get
there?
started with this?
Yeah,
You want to have some fun with this there, Kyle?
no, this is a
No.
no fun zone now.
Well, you just went and worked out. Come on.
Nah, it was just cardio. That's like hard. That's barely constitutes working out.
You're just all high on endorphins now, right?
Oh, shut up, Kyle. I walk 25 minutes a day, and I call that exercise. Now, you be good. That's what lets me to eat any sugar at all.
Oh, but
People
you're not
must be
really
laughing. This must not be that bad, huh?
At one point in my life, I was eating 3,000 calories a day and losing weight.
Yeah, I did that too. Actually, I think when I was in high school, I had a less than 2% body fat.
It's pretty good.
I grew 13 inches in 1 year.
Oh my god.
Yeah, I have an insane metabolism, or had. So I remember we went out to eat, and I ate like a full meal, and I had this huge dessert and all that. My parents were just floored, and I think it was like 4,000 to 8,000 calories. But anyways, not anymore. I'm borderline diet. Well, I'm right on the edge of diabetic.
Jeez.
Well, you know, age catches up with you.
That's true.
So if I walk 25 minutes a day, my body's really happy.
Ah, I gotcha.
And I call that exercise. So now that we've walked that side tangent, here we go.
Well, hang on. Whoever's got your Excel up, let me.
That's me.
Alright, let me do just so you can keep moving around. Alright, thanks.
What would you like to look at? Do you have allelic effects and average effect of allelic substitution done?
No, but I just took a screen cap. So
Fair. Did you calculate your P and your Q?
yeah, we have them from calculating.
Okay. Do you have your A and D's?
Hang on, I gotta move my file over. Say one more time. Oh yeah, A's and D's
For A's and D's, yep. Okay, so here's that's the formula for that.
Oh, hang on.
Well, the formula is right here.
Yeah, so it's
just what we found.
Yeah, it was in the same notes. Quantitative genetics, page 5 and 7.
Yeah, and Michelle, you know, bullied me into putting the alpha symbol instead of leaving it as just an A. So much academic bullying.
Yeah, that's pretty screwed up.
Yeah, no kidding, huh? Anyway.
I'm sorry.
You said 5 and 7?
Yeah.
5 and 7.
I think the equations are on 7, but it goes back and references page 5.
I see what you're talking about. Average effect of the. How where are you guys up to now? You on question 2 yet?
We're on 1H. We're having some issues, so a fresh opinion would help.
We have insane values for additive variance.
These plants are gonna explode. They have so much potential.
Okay, yeah. Let me catch up.
So, anyways. Well, I am happy with the fact that regardless of which formula we use, we get the same number. And I think the differences between the numbers is just coming from wherever the parentheses and symbols are. But yeah, that's the one thing I didn't notice on 5 is that if D equals 0, then if D equals A, then well, what if it doesn't do either? So that's interesting. And to be honest, with the notes, it looks like if you go to page 6, where it's VG equals VAD plus VDOM, he uses this formula for additive, and dominance is obviously the smaller of the two. And I don't see anything else.
Just real quick for the average effect of allele. So your P and Q change depending on what they are, depending on like which allele you're calculating. Is that right?
Yeah,
Yep.
Well, I mean, so like in the line with A, like capital A on the chart, it's got P under, A and A, which makes sense, but it's got Q under the heterozygote. Or I guess here, hang on. I'll just do the example real quick.
Here, go ahead. Throw it up there.
Oh, you want me to throw it up there?
Yeah. I've gone as far as I can go, man.
And this could also mean nothing. Alright, can you guys see the right screen here?
Yep.
Mm-hmm.
Yeah, so there's a P there and a Q there, and then a P there, Q there. So do you use that P and that Q, like for this bottom one, and then that P and that Q for this one?
Yeah, that would be correct.
Okay.
But I don't know if that's what we did.
So we had just used, well,
Although, looking further down further, that other Q is going to be basically Q is anything that's not your homozygous, I guess. So maybe I'm just overthinking it. I'm definitely overthinking it here. Look here. So like your Q for your first one is 0.2, and that's because earlier in the example your A is, or your p-value for homozygous dominant is 0.8. So I was overthinking it. You guys, it sounds like you guys did the right thing.
Okay.
Hey. Any sort of reinforcement's good reinforcement. Michelle, did you guys have any question about the average effect of the allele substitution? Because I know you guys were kind of shaky, and I looked, and that's what it seems to be to me.
Uh-oh.
Yeah, that'd be me. Just a minute.
Nukes launched.
I mean, I thought it seemed like we followed the equation. The only thing I'm thinking is the alpha value, the same alpha value, because like that A value we calculated for average allele effect of a substitution probably isn't the A value we want to use now for additive well, it's the effect of the substitution, not necessarily the additive, right? I'm just looking, because he's referencing, for like the additive variance, he's referencing that table above it, and where he has AD and negative A, he's got it listed as a coded GD. No, that's I'll go through it.
Like, do we have to calculate more things first then? Like from this table where it's like the coded GV and the genotypic value and the breeding value and the dominance deviation.
Okay, for average allele effects, where the answer is 57.48 and 100.73, and then it's negative for the dominant, positive for the recessive.
My numbers are higher than that.
Higher than that.
Yeah.
I guess, is the A and the D... Did I...
So I had got the equation for the A and the D off of page 5.
Okay. Oh, I used the A and the D from way later in the notes.
Okay. Which, and I don't know if that's right, but I think that, like, those numbers, that A and D, is basically our coded genotypic value based on those notes. Right?
Yeah. I think I'm gonna log off because I was gonna do some studying for 523.
Oh yeah, it's already after 9:30.
Time flies.
Oh yeah.
Well, that's a lot of work in one day. That's like super close to being done question 1.
That is so depressing to put it that way.
But there's 3 questions, not 9.
That's true.
And it's only Tuesday.
Yeah.
And we still have 2 days of lecture where he'll hopefully help us.
Maybe. Or just talk about theory.
Yeah. Hope it's the first part.
Oh yeah, he was going to tell us what he learned this semester.
I totally spaced out today.
I kind of did too. You got part of it.
Also, before you guys go, for the genotypic value, that's just the average count we had per genotype for that loci, right? As we used.
Yeah, like the one. The numbers.
Like 150, 163, 383.
Yeah.
Okay. Cool.
Right? Yeah.
Yeah.
Oh, okay. I see. Alright, how about 172 for effect of the first allele?
For big A, little a.
Yeah. Oh, for big A positive, for little a negative?
I had different numbers for them. Because one of the equations used Q and one used P. And I can share what I had.
Oh, you know what? I clicked the wrong one.
Okay.
Why for my starting? Hang on. So one of them is 172.32 though, right?
Yeah, I had that for the little a.
Little a. Okay. Yeah. Let me see here. Big A 114.88.
Yeah, that's what I got.
All right. Cool. Thanks.
Yeah.
And then 151.27 and negative 64.83.
Yep.
Sweet bear wine. Effect of a allele substitution. So for G, did you guys just have one number for loci A and one number for loci B?
Yes.
Yes.
Cool. Catching up.
So fast. I like that in question 2, I and H are flipped. So when I went ahead and posted the questions, I had to put them in the wrong order, just because that's what everyone's sheet would say, so now everyone has to keep them that way. GIH.
D. And then average alleles were 287.2 and 216.1.
Yeah. Although we're using that as the A value in the next step, and that's what completely messes us up.
I see. A plus D times Q minus B. Yep. A times Q minus B. Okay. Quantitative genetic variance. Is that and that's the same thing in notes?
No, it's in the variance, covariance, and heritability. So the only thing I'm looking at right now, so like on that quantitative genetic methods or models, page 7, like the alpha big A minus alpha little a, it also says should be equal to A plus D times Q minus P. That doesn't equal.
Page 7, you said?
On the quantitative genetic models, yeah, page 7. Right under average effect of an allele substitution, the alpha equals alpha big A minus alpha little a equals A plus D Q minus P.
I think I'm getting dumber. Why can't I define this?
The quantitative genetic models? Page 7. Yeah, sorry, I switched back to what you're just finishing, the average effect of the allele.
I gotcha. Okay, so you're saying... Oh, the A plus D, Q minus B, yeah.
Yeah. Because if you did A plus D, Q minus P instead of, like, our effects, that would give us something much smaller.
Wait, A plus D times Q minus P?
Yeah.
That's what I did to get those numbers.
But we multiplied it by Q or negative P also. Right?
Oh, I see what you're saying. I see what you're saying.
Yeah. I don't know, I'm just backtracking because if we have 287 as our allele effect, then we end up with like that 39,000 whatever.
Mm-hmm.
Which, there's no way that's our additive.
I'm doing frickin' IT help for my father too, over the phone with printers. No, this is, I was trying to show you, I got a 125.9 now. Using that formula just by changing my parentheses.
Oh.
And that's
How'd you
a
do
little
that?
bit. Well, I put it up there for everybody in the world to look at.
Which formula? So where did you get that formula from?
That is on page 5 of genetic variance, covariance, and heritability. If you look in the middle, you'll see that one formula that's yellow. The equals 2pq alpha squared. It's the formula right underneath it. The equals 2pq A plus D parentheses Q minus P squared. So you see that, Kyle? Right in the middle of the page 5.
Oh, yeah. See, you said yellow, and I'm like, I don't have anything yellow on it.
Really? Mine's yellow. He highlighted it.
2pq A squared?
2pqa squared, right underneath that is the formula.
2pq, and then in parentheses A plus D. Okay.
Yep. And the thing is, we did it also with the 2pq alpha squared, and it had the 39,000 number. So I got to work on this, figure it out.
Gotcha. Alright,
But anyhow, yes, with this parentheses and matrix setup, I've gotten 125.9, which is, I think, closer.
Yeah, I think that seems probably realistic.
Seems real.
Yep. I don't like the 2pq alpha squared one because D does not equal 0. I'm just getting rid of it.
Yeah, no, I think you're right on that.
So that would be V add for A, possibly.
A plus D, and that was our average allele sub effect, right? Yeah.
Yep. So yeah, I like these numbers better. 125.9 and 95.2, essentially.
That's, yeah,
So
more similar to what we've been getting and what you would expect to see.
so where did you put your parentheses? Sorry, I keep moving
Yeah.
my cell right in front of your
There you go.
screen there.
That's how I set it up to get it to work.
So
And the more parentheses I had in the past, the screwier the numbers got.
so you put it in between. Oh, see, I guess I also took the lazy way out, didn't I?
What's that?
I instead of typing in that P6 plus P7 thing again, I just clicked on my answer for average allele sub effect
Okay.
because that's what that is. I'm lazy.
No, it's minus, not plus. The average allele effect of the allele. Oh, forget it.
Yeah, you see what I'm saying? That's like that same basically little.
Yep.
That's weird though, because I moved my parentheses to the same. Oh, that's a new number. That's one that no one else has said yet.
What's
That's
that?
nice. I got 19,004.
Oh, I think we had that at one point in time, and then we went up
did
to
you?
36. Yeah, like I said, it's real funky.
A lot of crazy stuff has been happening.
You got to be precise with this one.
So it's squaring.
Oh, I'm doing IT again. I'll be back.
Times that.
Hello.
Mm-hmm.
Yeah, I don't.
U times P times Q times
Alright. Watch Excel just work for me now.
plus D. Is Jacques psyching you out?
Huh?
Is Jacques psyching you out? That's why you think it'll work now?
Is Jacques psyching me out? Is that what you said?
Yeah.
No, it's just, I don't. Because
I'm psyching Kyle out totally. I am totally psyching him out.
He's in my head! No, I just. Doesn't make any sense why that would not work like that.
Times. Ooh, not question mark. Tiny hat. Oh no, is my keyboard in French Canadian again?
I hate when that happens.
Does that not happen to Americans?
No.
Oh.
Hey, I got another different number.
I got 51.8.
I got negative 529.
Two times.
Because my parentheses aren't right. Hang on.
And then A plus D times bracket. And then Q minus P times hat 2.
I'm so confused.
Yeah, me too. How
Now I have 146,000.
Ooh, I know what I did. I think.
minus P. That's A.
125.9328.
Oh, that's a plus. That's not tax. Oh, I'm back to my original thing.
Now that just put this here, but change these. Oh, yeah, that worked real good. Okay, I got the same additive values as you now, Jacques. They might not be the right values, but they're
All
very.
right, I'm back. I'm done playing
Oh yeah, I forgot you were
IT
talking.
specialist. Oh, I just popped on. I'm doing 3 things at once. I'm multitasking, if that's what you want to call it. Anyway.
No, that's cool. I got the same values as you.
But so, what was this thing we don't do in America that you were talking about?
Have your keyboard accidentally switch to French Canadian? I don't know why it happens, but then certain things on the keyboard don't show up the way you think they are. You try and put it something, and it'll be a question mark, and you're like, why is this happening? But it's because your keyboard switches to French Canadian.
No, I don't have it switched to French Canadian.
So,
Unless my kids are playing April Fool's Day pranks. My daughter once changed it over to Cyrillic. That was
Very
funny.
annoying.
Anyway.
is that what they make pots and pans out of?
Yeah, dead Russians, that's what they do.
What
you
is
got 125, right?
125.9328.
I somehow got that.
Cool.
Yeah, I gotta admit, it's a little bit of witchcraft with all these parentheses. This is one of those weird Excel things.
So I am gonna say, to get that, you're technically the exponent's not written incorrectly.
Yeah, I see that too, because it's supposed to be the square root of this mess right here, or the square of this mess, right?
Yeah, the whole.
Yeah, you're supposed to square this whole thing right here
Yeah.
times
Although
that,
that's what. Wait,
and
that's
then it goes
Oh, yeah.
astronomically crazy.
just one parenthesis. Huh?
Well, yeah, you're telling it instead of it squaring q minus p,
Yeah, you're squaring the whole.
you're squaring the entire thing, which is what you're supposed to do.
It's. I guess you could
Why?
say it's exponential.
Yeah. And then whatever this number is, which is a fairly large number. P minus q is, or q minus p is a minus number, so it's minus 0.2 times, let's see, times d. D is a negative number, so that's why 0.2 times 103.5. I don't know. That's where I came up with a reasonable number. That's the best I could do.
I mean, seems pretty reasonable.
Yeah, but it seems wrong.
Does it seem wrong? Or does it?
Well, it's
does it
wrong
seem
because
right?
no. I mean, you're right. I was messing with the formula to get the number. Fine, we've got a reasonable number, but it still doesn't follow the formula. So it's wrong.
I mean,
You need the parentheses. You have to do it that way.
look, if you don't tell, I'm not going to tell.
Yeah, well, the other option is to do this. And you still get that. And that's about as simple as it comes for that formula. And I'm trying to say,
Do you think the notes are typed wrong?
no, although it's funny that we have to ask that. What I'm wondering is 103.5 times negative 0.2 is 20.7. And alpha, well, that's it. Alpha is 266.5. That's 287.2, and then you square that, and you're at 82,000. And then 2PQ being a small number is the only reason why that goes down.
doesn't seem right.
0.48 of 82,000. Yeah, it doesn't seem right, but that's, that's our, the A value is 266.5. Unless we want to reduce that down somehow.
No. So I mean, that's just what it is, man.
Yeah, the A value, that's the number of cysts, the average number of cysts, right?
Yeah.
Or did we calculate that wrong?
No, that's right. That's how it said to calculate it in the notes.
Yeah, so, by cyst, by average number of cysts, that's the genetic potential. The additive.
Sorry, how did you calculate the A?
Right there.
Oh, okay.
Yeah. I can't think of any other way to do it.
Wait a second.
If you have a better idea.
That's because A.
Okay, here we go.
A is Y of AA minus the average of the two homozygotes. Dang it!
What?
I was looking at the notes.
So is it screwed up in the notes? Or
I think it was screwed up in my eyeballs when I looked at it.
so this formula should instead be YAA or minus.
Yeah.
Before I miss that. So
Oh, that dropped. Okay. That changes some things.
oh yeah.
It does. It has some trickle down.
So that's
effect.
minus the what? I didn't catch that. Minus the heterozygote?
Here, let me. So it's the homozygous. Oh, you want me to just pull mine up?
Yeah, I just want to make sure I get the formula right.
Yeah, it's okay.
Michelle has infected my brain.
I'm sorry.
Oh, don't be sorry. It's the right way to do things.
All right. So it's your
Yep.
whatever you call that.
Your
Minus the average.
dominant hobo minus the average of the two.
Yeah.
You're
You
right.
don't-
I had the MH value.
No, I did the same thing. The only reason I realized it is because I scrolled up and looked at the notes, and I was just like, Kyle, you freaking dingus. And then this is also different.
So all we were missing really was
Just a heads up, this is also different. No, it's not. Never mind. We did that right.
so 116.5 minus. Okay. Wait.
Hang on.
Why is yours 279.5 for AD?
Because that's what it is, isn't it? It's your.
Wow.
Heterozygotes.
That makes a
Minus
difference.
Right?
Oh, monsieur.
What?
I think we swapped our freaking formulas is what it is.
Who's we?
Those of us.
Blame me.
Oh, I am
I
not
think I was
blaming
sharing
you.
at the time.
Michelle!
How could you?
So now everything's a negative value except for alpha E in like the lowest corner.
Sure.
Like
Don't look at this. Don't look at that.
it's like minus 116.5.
Yep.
Minus 103.5.
Yep.
Minus 38.32.
Yep.
And then 57.48.
Yep.
And
Wait,
then-
that's for your.
A's.
Oh. Yeah, I don't have that.
A, D, and then alpha big A, and then alpha little a.
AD.
Whoa.
So AD. Here, I'm just gonna actually, let's just actually do this. So it's this minus. Oh, okay, computer.
Who knew we'd be having this much fun on a Tuesday night?
I never doubt it.
Pretty freaking wild, man.
Then those average allele effects change.
So minus.
And now those additive alleles, well, they still might be wrong, but.
Let's just.
Okay, so what's the formula for D? It's Y, it's your homozygote minus your two heterotes together divided by 2, right?
Yes.
And A is your dominant homozygote minus your both homozygotes divided by 2, the average of them.
Yes.
Okay.
For some reason, it's not calculating this right, because, like, so I have heterozygote minus, what that would be, and my computer's like, nah. Like, okay, Excel, great program. Yeah, that just, thanks, Excel.
Yeah, that's what it should be. That's what it was before.
This.
Well, I haven't had to change the formulas for D. Just for my A. I keep wanting to call it alpha. It's not alpha.
Yeah, my Excel freaked out though when I did
Yeah,
that.
I know. Well, considering what we're doing with it,
Freaky stuff.
I'd be freaking out too.
Nah, man, that's not an excuse. I'm gonna drive up to Bill Gates' house and be like, "Listen up, nerd."
And he's going to be like, dude, how'd you get this address?
It's not acceptable. Alright, so average allelic effect. See, now those numbers make a lot more sense.
Yes.
Yeah. I'd buy that. If I read that in a published paper, I'd say, yeah, okay.
Average A effect minus 57, 173, 43. Yay!
Wait, you say negative minus 57?
No. No, I said minus 38.
Okay. I'm picking up what you're putting
95, 143, additive genetic variance. Okay, 4405 still seems a little large, but minus 56 makes much more sense.
Yeah.
For your additives?
But that's because we haven't changed. Yeah.
So we need to take out or move the bracket with the exponent 2?
I mean, you know what? Here, let's just put yet another pair of parentheses in here and see what happens. Okay.
Yep, that's what happens.
Makes it big again.
So the question is...
Wait, for average allele sub-effect, did we get negative 95, negative 143?
Mm-hmm.
Yes.
Yes. Yet again, I've got to change everything.
That's why I just left all my equations in.
I should have just left them all in the spreadsheet and been like, reference attached spreadsheet.
Well, there's that too. I do that, and he seems happy with that. So.
It's probably more legit that way because then he can see that you actually have like live equations and you just
Yep, I think that's what he likes.
a bunch of screenshots.
I have to say something to my son here real quick. Too legit to quit. Oh, okay. Anyway,
That was adorable.
I'm terrorizing my kids with the '80s music, but. So are we going to try and go with 57 and 56 and fake our way through it or what?
For the additive genetic variance?
I mean, I like the number, but I hate the fact that it doesn't match the formula.
They don't always match up though. Sometimes the formulas that come from the text are really complicated, and what we end up using is a simplified version.
It just bugs me. Well, what is it telling you in the text? It's telling you to square the whole dang thing. A plus D times Q minus P. You square the whole thing.
So if we squared minus 57, we would still get a reasonable number.
Well, here, I'm gonna try something here, just because I'm a dork. Apparently, well, you know how it is. You can't just let things go.
Not on a Tuesday. On a Saturday, it's a little easier.
I don't know. You would not have to try and convince me very hard to like just let some stuff, let this go. Like, Kyle, you should just go with it. Like, okay.
Okay, so I did the other formula, which is 2pq alpha squared. And it wants to be 4405. I feel like...
You think it might be 4405.
Is our alpha correct, is what I'm saying. 4405 is... I can't even fathom that for freaking additive potential.
And that's
additive variance.
1,405?
Yeah.
Yeah.
And the only way to change it really is our alpha.
So, no. So that might be right. Because our phenotypic variance is 10,527.
What? Where'd you
I just did variance of just that inner square, and that's what I came up with.
You did the variance of what? Sorry.
Of just the average counts. Because technically that would be similar to like an entry mean. I mean, right?
You mean just over? Did you do it on a different file or?
Nope. I just did it in the middle. Just
the 9 in the middle.
Yeah, the average counts per genotype.
Why is our variance so high?
I don't understand how you got a huge number. Sorry. Is that a VARS that you did?
Mine only has a VAR.
Yeah, I just did VARS of that, and it's
Yeah,
10,527.
Okay, you know what?
It's nice.
Yep, we're back in business, baby.
YOLO.
So we're moving alpha? Or no, alpha's fine, we're moving brackets.
Yep, we're making it right. That makes my B allele at 8697. How do you like
Yep.
that?
Yeah,
Got the same.
No, it wouldn't let me change it.
makes sense.
I'm willing to go with that since it's an insane. I can't get that. How you can be 377 to 31? I get that there's a variance there, but a variance of 10,000? That just boggles my mind. That's the part of math I don't really get.
So what's the equation now? For additive.
It's
Right here.
Jocks, you got it.
Yep.
2pq blah blah little bracket. Ah!
Did you switch over to French again?
Nope. Then I would have shouted tabernacle, which I don't completely understand, but it is offensive.
Tabernacle?
Tabernacle.
Tabernacle.
I think like it's kind of equivalent to fuck, but you can't use it in the same way that you can in English. So like you can't tabernacle someone's mom.
Whoa.
Okay.
Well, let's find this out.
I don't understand it. I think the open French Canadian first truly makes sense. Okay, so you guys have 44,005 and 8697.
Oh, here we go.
Yeah.
Tabernacle. Is one of the worst curse words you can use in French Canada.
That's awesome.
It's a sacrilegious word, which takes the word tabernacle in vain. Ooh, you nasty girl.
How do you, how do you spell this?
TABRNAC or NAK.
Like the Tabernacle Choir.
Yeah, the tabernacle is where you keep the Eucharist.
I'm gonna.
And so taking that in vain is going against French Catholics, Canadian French Catholics.
I just added my Canadian friend and just said tabernacle. I'll see what he says back.
Yeah.
Hey, good news. Dominance variance seems like easier. 2
What?
PQD all squared.
Yeah.
An easy equation. Fancy that now, would you?
Well, it's technically
E times Q times D.
technically we can use. Watch this.
We got a hot shot.
I'm not. It's not. I'm using this. Try not to be mad at me, Michelle.
What? Am I gonna be mad?
What? So we could use this simple equation instead of this bigger equation and get the same answer for our additive. Just saying.
Yeah, you see, you could do that.
But why? We want to see what's going on behind the curtain.
We have grown as people by doing it this other way. And Sean,
I feel
I'm
like
just trying to minimize that. I feel like he's trying to cheat us out of our accomplishment.
I feel like an idiot. So.
You can't take away my creative desire to explore.
Is that what we're calling that now?
Knowledge is power, man.
Are you getting a 5141?
What?
0.
Wait, for your things.
For
Dominance?
for VDOM. Yeah.
Oh, I got 2468 and 333. Plus
I got the same.
What do I?
Uh-oh.
What's wrong with me?
2 PQD?
Yeah.
Bracket 2 times.
2 PQD squared.
Yeah.
Am I hot linked into the wrong
No,
thing?
no. All of it squared. So multiply all of it together first and then square it.
Okay.
Oh, okay.
Yeah.
That's Q. That's D.
Well, that means that there'll be a difference. And what I mean by that is... Ooh.
I got that. I got what just showed up on Jock's screen too.
What did you get?
2,468.1024.
That's it.
Excellent.
333.7929.
That's that shit I do like.
Oh my gosh, I can cry. I'm so happy.
Well, if it makes you feel any better, there's only 2 more questions left.
Excellent.
And this was the easy one. It took us like hours.
Yeah, remember, this is the easy one.
I think we make things harder than they need to be.
It's true. We ended up doing most of these like 3 or 4 times.
Yeah.
Yeah.
For practice.
So everybody's got this, huh?
Mm-hmm.
Okay.
For now. Until someone convinces
For
us.
now.
Yeah,
Well,
I feel good about that.
Someone will be like, oh, but what about this? And then we'll sit there and and haw and...
My friend said back, Sacre Blue.
Oh, what an educational experience.
I'll ask my daughter, Lily, about using that word, and she'll probably be really mad at me.
Stop with me.
Although she's pretty anti-Catholic, she's a brat that way, so. I'm Catholic. And she's all the horrible things the church has done to minorities and women and all this, and I'm like, yeah, okay, fine. What do you expect? Why do I have to pay for their sins? Anyway, So I'll tell her that either she'll be super offended or she'll think it's hilarious. I don't know.
You never know. Teens.
Holy cow! We're actually done with a question on Tuesday night and feel confident about it? That's so odd.
Shall we throw in the mouse and call it a night?
I think
Yeah, that's
so.
fine.
I did not realize it was this late already.
It's late.
Yeah, neither did I.
Past my bedtime.
Yeah, I'll be back online tomorrow and ready to crush out some data crunching. So
Sounds good.
I might be on later tomorrow, but
Yeah, Emily and I have a study session at 7 with the other class, but aside from that,
Michelle can charge forward and let you guys know how our wheel spinning went,
Yeah.
or maybe just our sheer charging forward.
well, I was going to say it's at 7, so I'll probably get on here before then and then jump over and see if anybody shows up and jump back in.
Sounds good.
all right.
Have a good night, people.
Have a good night.
See you guys.
Have a good night. See you guys.